Okay, welcome back, everyone. I see my co-moderator, co-chair, Dr. Bernie here. So that's our cue to get started. It is 10:30. So we are now going into the second session of the plenary, morning plenary. And very pleased to introduce our next talk, which is titled Potential Barriers to Accessing Care, Is Help on the Way? And today's talk will be given by two of our patient partners, Risa Mallory and Anne McAllister. Risa is a retired psychotherapist and unfortunately had a serious cardiovascular event, and as a result has become very passionate about heart health advocacy and is really uh, focused a lot on establishing collaborative medical care and helping with knowledge translation. She volunteers both on a local, national, and international level with healthcare organizations and patient advocacy groups. So we're very fortunate to have her today. And then also Anne McAllister is a retired lawyer who spent a number of years in private practice before working for Justice Canada, retired in 2010. And at the time, she was also unfortunately diagnosed with a rare cardiac condition. And since her retirement, she also has become very active in advocating for research around rare cardiac conditions. She's also very giving of her time and volunteers actively at the University of Ottawa Heart Institute. So we're very thankful to have her today as well. And then finally, Dr. Edgar De Silva is our fellow in rare cardiac conditions. He completed cardiology and echo training in Brazil, and I'm probably going to butcher this a bit, Edgar, so at the Universidade Estadual de Campinas in Sao Paulo, Brazil. And his interests are rare cardiac conditions and cardiac imaging. So welcome to all three of our speakers. And with that, we're first going to hear from Anne McAllister, who's going to tell us about her patient journey. Anne, are you able to unmute and you can display your video now?
That's okay.
Yeah, we hear you now. Perfect. Yeah, and we see you as well.
Okay, great. Okay. Good morning, everyone. I've been asked to start this panel off by relating my experience as a patient diagnosed with a rare cardiac condition. In March of 2009, I was diagnosed with cardiac sarcoidosis. When I was diagnosed, I was 58 years old, practicing as a lawyer in the government. My story, however, started 2 years earlier, or two and a bit years earlier, in the fall of 2006, when I experienced repeated episodes of tachycardia. I had no previous heart issues. My only significant health issue was thyroid cancer when I was 26. My only regular medication was Synthroid. I had no physical limitations. I worked hard at a demanding job and was relatively active and traveled extensively. My story may be a bit out of the ordinary in that it all took place in a hospital setting. I did not go first to my primary care physician with symptoms, followed by a referral to a cardiologist. As I mentioned, in the fall of 2006, I had several trips to the ER with tachycardia, where I was treated with adenosine and given a prescription for beta blockers. Medication failed to manage the tachycardia, and after quite a few trips to the ER, I was eventually referred to the specialists at the Heart Institute. I had several appointments with Heart Institute cardiologists. My medications were adjusted, and I was ultimately put on a list for an ablation. This was mid-December of 2006. My Christmas Eve, however, was interrupted by another trip to the ER with an episode of tachycardia. A few days later, just before New Year's, I returned to the ER, this time with bradycardia, and I was admitted to the Heart Institute. A pacemaker was implanted during the first week of January of 2007. As far as I know, no further investigation was undertaken to determine an underlying cause of the arrhythmias or of the sudden change from tachycardia to bradycardia. The assumption seemed to be that the pacemaker would resolve my cardiac issues. So we go forward now 2 years to early winter of 2009, when I found that I was really out of breath going upstairs. I had difficulty walking on snow-covered ground. So I went to ER again, where I was told that I had symptoms of heart failure. I was admitted to the Heart Institute, where a diagnosis of significant heart failure was confirmed. I was discharged a day or two later and told that I would be followed as an outpatient. Over the course of the next couple of days, I really felt that my symptoms had worsened. So I returned to the ER and was readmitted to the Heart Institute. It was at this point that questions were asked, not only about the relatively sudden onset of heart failure, but also about my previous history of arrhythmias. The team on my case included electrophysiologists and heart function specialists. A respirologist was also brought in for a consultation. Various tests and scans were undertaken. A range of potential causes of my cardiac issues were ruled out. And eventually, by a process of elimination, cardiac sarcoidosis was raised as a possible diagnosis. There were several reactions to this. The cardiologist who had discharged me from the Heart Institute earlier in the week scoffed at the idea and said that I did not fit the profile for sarcoidosis. My electrophysiologist said that he had never encountered cardiac sarcoidosis in the course of his career, but the heart pulmonary specialist had familiarity with the disease. A diagnosis of cardiac sarcoidosis was confirmed. It had caused damage to the heart muscle, hence the heart failure, and in hindsight had caused the arrhythmias 2 years earlier. My immediate reaction was shock that I had a rare cardiac condition, and the cause of the condition was unknown, and that there had been significant damage to my heart. I was told that sarcoid could appear in other parts of the body, such as the lungs or the eyes. My first thought was that the disease could spread, that the quality of my life might deteriorate, and indeed the length of my life might be shortened. This immediate reaction, though, was mitigated as I talked with my doctors and as the focus turned to treatment. I was also very much comforted by having the opportunity to read materials on sarcoid that were provided to me by one of my physicians. He seemed to know that I like to read things, and so that was very helpful. In terms of treatment, I received the standard of care for sarcoidosis at the time, a course of prednisone over several months, which I tolerated with only minor side effects. My implanted device was upgraded to be a pacemaker and defibrillator. I was referred to a respirologist and an ophthalmologist so they could assess whether there was any sarcoid in the lungs or the eyes. I responded well to treatment, and sarcoid was declared to have burnt out within a reasonable period of time. Throughout this time, however, I did feel vulnerable and fragile, as my future did seem a bit uncertain. As follow-up scans confirmed no sign of sarcoid in my heart, and my respirologist and ophthalmologist found nothing of concern. My natural resilience took over and I regained confidence and a positive outlook. In time, I retired. I resumed traveling widely. I worked very hard at lowering my golf handicap, and I became an active volunteer at the Heart Institute. I am now followed by a heart function specialist and have my ICD checked on a regular basis. My condition has been very stable for a number of years, and there has been no sign of a flare-up of sarcoid in my heart or elsewhere. I have considered myself very fortunate to live in the same city as a world-class heart institute. I had access to everything the medical system had to offer, yet my diagnosis was delayed for 2 years. That was 16 years ago. I am confident that today, in light of the work that Dr. Burney and his colleagues have done and continue to do in regards to cardiac sarcoidosis, that this would not happen to me today. But over the years, I have spoken to a number of patients across Canada who have really struggled to have access to timely testing, timely diagnosis, and timely treatment. Everybody's story is different. Mine was a fairly straightforward story. Others are more complicated. My fellow panelists will consider systemic barriers to diagnosis and treatment and will suggest strategies for breaking down some of these barriers. I would like, in conclusion, to leave you with a few general questions to keep in mind as you listen to the following presentations. What practical barriers exist to timely diagnosis and treatment for patients with rare cardiac conditions? What measures can be implemented now and in the future to increase the likelihood of timely diagnosis and treatment? How can awareness of rare cardiac conditions be raised across the medical profession, from primary care physicians to cardiologists and other specialists associated with large hospitals or medical research centers? And finally, what steps can be taken to engage policymakers and funding entities to eliminate barriers to diagnosis and treatment? Thank you.
Thanks, Anne. That was wonderful. Next, we're going to hear from Risa Mallory, who will give us a patient's perspective on these barriers and how potentially some inkling of how we might start to overcome them.
Thanks, Ian, and good morning, everyone. In healthcare, telling our story isn't just about sharing our medical journey. It's about amplifying patient voices to drive meaningful change and humanizing healthcare experiences through our very authentic narratives. So thank you so much to Anne for doing just that and for all the advocacy work you do. So for the audience participation part of this day, we are going to launch a word cloud. While we're doing that, if you could reflect on Anne's story, please access the QR code if it becomes available to us and enter a word that describes barriers to care from a patient's perspective. There's the QR code. So you want to type in a word that is a barrier to care that comes to your mind, either from Anne's story or that you're aware of from either your clinical or research experience. As you enter your word or words, we'll watch the cloud generator do its thing, and this will take a few moments. So feel free to, there we go. Distance is one, good. Dismissal, that's a good one from a patient perspective for sure. Good stuff. The more frequent a word is, frequently the word is typed, typically the larger it becomes. So right now we have a real breadth. There we go. So knowledge gap is getting bigger. Awesome. Give it a few more seconds. Okay, I'm starting to slow down a little bit. Knowledge gap seems to be the largest one that's been reflected in your answers. So thank you very much for doing that. We'll go back to the slide deck now. So I'm going to elaborate on all of the ones that were mentioned and maybe add a few more over the next few minutes. First, I'm going to discuss the barriers to care with respect to rare conditions, and then the barriers through the lived experience, as Ian mentioned. Before I begin, you'll notice on this first slide of knowledge gaps, just wait for that slide to appear. There we go. That there's an asterisk beside the second point. This identifies a specific barrier that Anne experienced or continues to experience during her rare condition journey. I'm hoping that this will humanize the rather dry points that I'll be speaking to. And please take note of any other asterisks you see throughout my talk, as these are also Anne's experience. So let's get started. Patients may not experience symptoms or understand when to seek care. Healthcare providers may lack training on rare cardiac conditions, and policymakers and industry leaders may not be aware enough to have a vested interest, and so the knowledge gap is there. With respect to delayed or incorrect diagnoses, many healthcare professionals may not be familiar with rare cardiac conditions, leading to misdiagnosis or a prolonged diagnostic process. Patients may need to travel significant distances to access cardiologists or centers with expertise in their disease. And rare cardiac conditions often present with nonspecific or overlapping symptoms, which is complicating a definitive diagnosis. Psychosocial challenges may arise because of uncertainty and isolation, which can be associated with rare diseases. Patients and families struggle to find others who understand their experiences, and family members, who often play a key role in care, can lead to burnout. Inadequate research is a result of policymakers and industry lacking interest in funding research for rare cardiac diseases. The scientific community may lack the initiative to engage in this research because it may not be, the sexy topic of the time. There is obviously a smaller pool of patients to recruit as research participants and also as patient partners. And so as a result, it is challenging to identify optimal treatment approaches when the data is limited. Expensive technology may not be available even in larger centers. Few healthcare centers specialize in rare cardiac conditions. We're very fortunate here in Ottawa and other places in Canada, and therefore treatment guidelines may not be well established. And again, the lack of research on rare cardiac diseases hinders the development of best practice treatment. So systemic inequalities lead to patients in rural and underserved areas facing greater barriers to accessing their specialized care. More obstacles facing non-native speaking patients or those from marginalized communities, and poor coordination between primary care providers, specialists, and multidisciplinary teams. Logistical challenges are evident because accessing distant specialty centers can be a significant hurdle for many patients. Carving out the time or getting alternative coverage for patients who are also caregivers to others, either children or elderly, or other sick spouses may be a challenge. Reasonable accommodations by employers may not be made for medical appointments or absences due to health. Limited availability of specialists may lead to long delays for appointments, and communication barriers may be related to either native language, cultural translation, medical jargon, or even a lack of patient-centric approach to care. Now we're going to talk about the consequences of these behaviors as experienced by patients and their caregivers. Some recognized again by asterisks that Anne herself identified on my slides. So physical health-related consequences can lead to delayed detection, resulting in advanced disease stages, I should say, by the time a diagnosis is made. This in turn leads to reduced treatment effectiveness and worse prognosis for the patient. Limited access to specialized care may result in the use of inappropriate or ineffective treatments, leading to poor disease management and complications. Without timely and appropriate interventions, patients face higher risks of complications such as heart failure, arrhythmias, or sudden cardiac death. And poor disease management can increase the likelihood of developing secondary conditions, such as pulmonary hypertension, stroke, or kidney dysfunction. Such psychological consequences are common because the prolonged uncertainty of undiagnosed or misdiagnosed rare conditions contributes to a patient's increased levels of anxiety, depression, and feelings of isolation. Anne mentioned that she waited 2 years for a diagnosis. Rare cardiac conditions often lack robust patient support communities, leaving families and patients feeling unsupported and misunderstood. Negative experiences with healthcare teams or institutions can lower a patient's confidence not only in the process, but also in the providers. These negative experiences, combined with low trust in doctors or denial of the severity of our symptoms, can lead to a lack of engagement and/or treatment avoidance. And the complex care needs of patients with a rare cardiac condition can place significant strain on the informal caregivers and family members that we mentioned before, leading to burnout and financial stress. Financial constraints can be experienced as a result of barriers to care. Patients can incur expenses for specialized testing and travel to centers of excellence. Chronic illnesses may prevent patients or caregivers from maintaining steady employment, exacerbating any financial instability. Limited insurance coverage for rare diseases can force patients to go for life-saving force patients to pay for life-saving interventions or forgo care. I have some personal experience with this point. I had a complicated triple spontaneous coronary artery dissection while spending the winter in Arizona. And although I had purchased third-party health travel insurance and my claim was for 400,000 Canadian dollars, it took over 13 months to settle this claim. And this long adjudication period, coupled with fielding weekly calls and mail from American collections agencies, was very stressful. Patients' quality of life can suffer greatly as a result of barriers to care. Persistent symptoms such as fatigue, breathlessness, and pain can severely impair our daily functioning and reduce a patient's quality of life. Physical restrictions due to cardiac conditions can prevent patients from engaging in work and hobbies or social events, and that's diminishing our overall sense of well-being. Dependence on specialized care and the uncertainty of the rare condition can reduce a patient's sense of control over our health and our lives, leading to feelings of helplessness and hopelessness. Inequities in care is a huge barrier for rare cardiac conditions. Patients in underserved areas from minority backgrounds or with limited financial resources face worse outcomes due to the compounded barriers to care. Geographic and systemic barriers create unequal access to diagnostics, specialists, and advanced treatments for rare conditions, resulting in poorer outcomes for certain populations. And my last slide, of all of the barriers discussed here can prevent patients from enrolling in or collaborating on clinical trials, reducing opportunities to improve treatment and the understanding of rare cardiac diseases. Poorly coordinated care leads to incomplete data, limiting the ability to develop effective therapies or inform policy changes for rare conditions. So quite a long laundry list. And so, Edgar, we're wondering, is help on the way?
Thanks, Risa. That was wonderful. So Edgar, you've got your work cut out for you. There's lots of issues. So why don't you tell us about some potential solutions to these issues?
First of all, thank you for inviting me to this meeting. It's an honor to work with all the amazing professionals in our rare cardiac conditions department. So I'll be talking about some initiatives to overcome barriers to care. I don't have any disclosures. So the current landscape in Canada is going through urban centers, rural centers, and Indigenous communities, and each one has its own problems with access to care. So some of the goals for good access to care are to provide equity of access to all the patients with rare cardiac conditions. They should be treated as all the other Canadians or patients with more common diseases. It needs to be a patient-centered approach, and there must be collaboration and coordination between governments, patients, and healthcare professionals, as well as researchers and the industry. Some of the local initiatives are community-based solutions, and they are basically related to healthcare education programs to educate professionals and patients to understand and learn about their diseases, promoting primary care services, community outreach to raise awareness of the diseases, maybe creating mobile clinics to have more access to services in rural communities, and providing financial assistance, as Rita mentioned before, as well as diagnostic testing and referrals to streamline and reduce wait times and treatment delays. More of the local initiatives are increase awareness to the family health services, improve access to mental health and specialists, as many patients have burdens of mental health, modernizing our health system with digital tools and information and referrals, all digitized, build the health workforce, creating more spots and have more people working in the system, investing in primary care education and training so people, especially the primary care providers, are more aware of the rare diseases, and also investing in information systems to make it easier for clinicians to work together with specialists. As part of the provincial plan, they're trying to expand specialized clinics, creating provincial centers of excellence where we have specialized clinics dedicated to diagnosing and treating rare cardiac conditions, and also enhance the referral networks to improve referral networks and facilitate timely access to specialized care for rare cardiac diseases, especially access to genetic testing. One example is the Canadian Center for Rare Cardiac Conditions here at the University of Ottawa Heart Institute, where is a place with a specialized hub offering comprehensive care for individuals with rare conditions. Specialized clinics at the Heart Institute provide tailored evaluation, diagnosis, treatment, and rehabilitation, and the center serves as a centralized point of contact, also streamlining the process of identifying appropriate clinics and facilitating the appointments with trained experts. Here is an example of our website. We created this website with a registry of all the clinics in Canada, so patient can have information, and also healthcare providers can have resources to understand more about rare conditions. We have all the clinics divided by province and cities, so people can have access to information and see where is the closest place that they could go for if they are diagnosed or they think that they have something related to rare cardiac conditions. Again, part of the provincial plan is to implement coordinated care pathways, increase funding for research, enhance data sharing across all the provinces. There's this program called Genome Canada All for One initiative to develop clinical genomic testing sites across the country that's going on right now. Provide educational and training, and also expansion of the telehealth to broaden services to reach underserved areas and ensure remote consultations so patients don't have to travel far to get access to care. Again, part of the provincial plan is to promote integrated approach between provinces. Just an example of this Genome Canada project, there is 13 million in federal investment through the Genome Canada and 26 million in co-funding from industry, healthcare organizations, provincial and other partners to promote more access to care in rare cardiac conditions and other rare conditions. It's also promoting public awareness campaigns and stakeholder engagement in planning and decision-making and the policies. Some examples of the provincial strategies that were successful, like Ontario implemented centralized drug formulary, Alberta focused on public awareness campaigns, Quebec adopted disease-specific eligibility criteria allowing broader access to patients to rare conditions. Manitoba created initiatives for patients' navigation through the system to streamline access. And British Columbia established a rapid review process for new drugs, ensuring faster access to treatments. On the national level, there is increased government funding for research on rare cardiac conditions to accelerate the development of treatments and therapies. More training specialists like myself, the first fellow in rare cardiac conditions here. So on the national level, they're trying to invest in training for healthcare professionals to specialize in rare cardiac conditions and also promote data collection and registries. There's a collaboration between federal and provincial government where the government of Canada made available up to $1.5 billion to provinces and territories through bilateral agreements to help to improve access to new and emerging drugs, as well as support the enhancement of access to existing drugs. There's the Rare Disease Research Initiative that will fund rare diseases research in Canada, and they provided $32 million over 5 years to advance rare disease research in Canada. And in March 2023, the federal government launched the National Strategy for Drugs in Rare Diseases, and this commits up to $1.5 billion over 3 years to help to increase access, affordability, and effective drugs for rare diseases to improve the health of our patients. And what is the future? So to continue to increase awareness about rare cardiac conditions among healthcare professionals and the public to improve early diagnosis and access, they're trying to create this pan-Canadian health data content network that will provide information flow for connected care and provide a future foundation for pan-Canadian standards, ensuring seamless and secure data exchange, especially between provinces. The government policies and funding initiatives should prioritize research and training to address unique needs of patients with rare conditions, and continue with the national strategy for drugs to develop a coordinated strategy to get patients the effective treatments they need. Here are some of the resources where all the information from this presentation came from. Thank you very much.
Thanks, Edgar. That was a wonderful overview of both local and provincial and national initiatives. With that, I invite our three speakers to come back online, and my co-chair. And we invite audience members. We have about 3 minutes left over for questions. So if anyone has questions, please type your question in the Q&A. I'd just like to pick up while we're waiting for questions on a couple of points that both Risa and Edgar made. And so I'm a healthcare provider for someone who provides care in someone with rare cardiac conditions, and I can tell you, I see that it's hard for patients as well as healthcare providers. That often we don't know where to direct patients. So for clinical trials that are ongoing in the country, we may not be a participating site, and it's hard to get that information about where are the sites that are offering these state-of-the-art clinical trials. And so in my view, we're in dire need of networks, and that's something that was highlighted by your talks. So the website that was developed here at the U of O, we actually recently received, we've been starting to receive communications from other patients across the country because when they Google, this is what they find. So we're one of the first websites that come up. And so we've now started to accept referrals from elsewhere in the country. So it just shows there is a need both on the clinical side and on the research side, in my opinion. And we really also need to engage patients to be involved with developing these websites to make sure that they're friendly not only for patients but also for for healthcare providers and researchers. So maybe I'd ask each one of you to comment on that, what your perspectives are. So I don't know, Anne or Risa, if you had any comments related to that about the need for networks and knowing both on the physician side and the patient side, where to go exactly.
From my perspective, I'm involved with the Canadian Women's Heart Health Alliance, and one of the initiatives they have going on is a registry, for lack of a better word, for researchers and patients to connect as such. So it's not specifically about rare cardiac conditions, but they do have just launched actually, so it's a fairly new initiative, and that's their goal, is to connect researchers who are looking for both patient participants and patient engagement folks to help them with their research and be subjects is not the correct word anymore, but to participate in research.
And maybe I can just add, from the beginning, I've been a firm advocate that, the approach to rare cardiac diseases has to be completely different to the approach to common diseases. Common diseases, what do we do? It's all about education and knowledge translation to make sure that, other doctors understand the disease. Here, it's nothing about knowledge translation. It's all about saying, you don't need to worry about this patient. This is where you go for specialist care and just send your patient as soon as possible. The example is there's data to show that a family doctor will see a case of cardiac sarcoidosis once every 20 years of their career. So they can't be expected to know. You as a patient will know much, much more than your family doctor. And you can't expect your family doctor. The key thing is for your family doctor or whoever to get you to a specialist center.
I agree with you. I think the challenge arises that the family doctors often don't trust a patient's assessments of their symptoms. And so, like our first speaker said, the patient case study she presented, the woman was dismissed because she was diagnosed with anxiety. And so that's an ongoing, entrenched barrier that a lot of people experience when they present to their primary care physician or to emergency departments with something that is rare, or they're outside of the box. They don't fit the mold. They're not the garden variety, as she mentioned.
Yeah, I do think the other barrier we have is access to genetic, and that's something we did a workshop on this in the fall, and polled other healthcare providers, and this is an issue across the country. So it's something we're working on locally because so many rare cardiac conditions have genetic components to them. It's, as we've heard earlier today, it's central to managing these patients and may help direct therapies as well. So it's very important to continue to work on that. So unfortunately, we're over time, so we're going to move on to the next speaker. But maybe if we have time at the end and there are questions, we can come back to this group. Thank you once again.
Yeah, we'll move on to our next talk, and I hope I don't mispronounce your name, but I think you're Professor DeHoon from the University of Nijmegen. Again, that may be mispronounced. He's a full professor of bioinformatics at the Radboud University Medical Center, dedicated to the advancement of personalized medicine approaches for rare diseases by integrated use of omics and clinical data. He's coordinator of the European Sympathic project, aiming for acceleration of drug repurposing for rare neurological, neurometabolic, and neuromuscular diseases. And very excited to hear his talk on. Very, very excited to hear your talk on drug repurposing in rare diseases.
Well, thank you very much for this kind introduction. Is the sound and the slides are all right?
Yes, perfect.
Okay, good. Well, thank you again. So welcome from Nijmegen, the Netherlands. This is a special occasion, of course. Rare disease, the day is celebrated across the globe, and it really give me delivering this lecture here in Ottawa really makes me feel this global connection. So thanks again. I have a special connection to Ottawa as well, because the Sympathetic project that I'm coordinating, Theory Institute in Ottawa, and in particular Hans Lochmüller's group is very much involved. So also there is a global connection in the rare disease field. So I have no disclosures that are related to any commercial entities, and then the work I will be presenting today is funded by the European Union. So Rare Disease Day 2025 in Nijmegen started actually with the patient. So we invited the patient to our work meeting that we usually have on Fridays. And actually we heard a lot about the patient living with their rare disorder and what it means for the patient. And that makes me and the team really work harder for the good cause. So this was Rare Disease Day, almost ended now in Nijmegen. A little bit about myself. I'm a pharmacochemist by training. I did a postdoc in genetics, then collected so much data that I had to study bioinformatics, in which I'm now a full professor. I specialized in myotonic dystrophy as a rare disorder. I will come back to that in my talk. And furthermore, I work at least 50% of my time on developing fair data infrastructure for rare diseases. And all of this is happening in the Myotonic Dystrophy Expertise Center in Nijmegen and Maastricht. So myotonic dystrophy is a rare disorder. It's a multisystemic disorder that is mostly known for myotonia as its primary hallmark. So the difficulty relaxing your muscle, so measured by long hand opening times, but it affects almost all the organ systems, including also the heart. So many patients suffer from a cardiomyopathy. So when I started in Nijmegen, I was very much inspired by our neurologist, who now just retired last year, Professor Basile van Engelen, who said to me, well, only 6% of rare disease patients have access to disease-modifying treatments. I know about all the developments out there, like genetic therapies and gene therapies, which we already heard more about earlier this morning. But rare disease patients do not want to wait for another 12 years until some genetic therapy may become available for their specific disease. So we need something between care, taking good care of our patients, and cure. And that's where I came in, and that's what the rest of the topic of this lecture will be on. So can drug repurposing mean something between care and cure for patients? And this is where we started. We started off with the optimistic trial, which was successfully coordinated by Professor van Engelen, in which they tested a lifestyle intervention called cognitive behavioral therapy, and actually observed that some patients responded well to the therapy and some responded less well to the therapy. So there was heterogeneity involved. And actually, we thought we could use this response to also identify drug targets, because the drug targets may actually respond to the lifestyle intervention. So we associated this response to drugs with possible lists of drug targets and to identify new and repurposable drugs. And this strategy is interesting because this repurposed drugs can actually either be used in combination with these lifestyle interventions, but perhaps also replace in patients where these lifestyle interventions are difficult to continue. This is the key figure from that research published in this article in BMC Medicine that you may want to explore later. But we measured RNA expression in the blood before and after therapy. And actually we observed that there are some RNAs that are positively associated with the disease severity, so are more up or down regulated when it comes in more severe patients compared to less severe patients. And we associated that with the response to the therapy. And when we looked at the intersection of those, we saw actually an anti-correlation in this graph. And that was very interesting because here in the left upper panel are genes that are downregulated and in the more severe patients, but are upregulated again in patients who respond to the therapy. And on the other side, in this lower right-hand corner, are genes that are upregulated in the more severe patients, but downregulated again in the patients who positively responded to the lifestyle intervention. And when we look at these genes more carefully, we indeed observed some drug targets, in particular in the retinoic acid signaling pathway, but also in the HDAC, so the histone deacetylase inhibitors, and some other druggable pathways like the lipoxygenase pathway, the tyrosine protein kinase pathway. And we are following up on these projects. And that was actually the start, we followed up on these drugs, and that was actually the start of a new project and in which we wanted to accelerate therapy development for rare diseases by focusing on symptomatic treatments using drug repurposing. Drug repurposing, also called drug repositioning, is the use of existing drugs for new indications. And this may actually accelerate the access to drugs by patients quite a bit. The regular drug development pathway takes at least 12 years and at least $1 billion or $2 billion US. However, drug repositioning or drug repurposing can be done at shorter time scales and with lower investments. So there are two ways on how you can look into a drug repurposing. You can either investigate known drugs. Often they are already off patent, but it also happens that drug repurposing projects start with investigational projects that are already in research and development with pharmaceutical industries for new indications. So with drug repurposing, we think that we can make a faster translation from preclinical work to clinical studies because most of the safety and quality studies have already been done. And it is therefore easier to get regulatory approval for a new indication. Well, there are some examples in the myotonic dystrophy field where I'm working. So metformin has been shown in phase 2 and now also in phase 3 trials. So metformin and anti-diabetic drugs to increase the capacity for physical activity of myotonic dystrophy patients. And there was also a recent trial by AMO Pharma on tidagliflozin, which was originally developed for Alzheimer and seems to improve also symptoms from myotonic dystrophy patients, although it didn't reach its primary endpoint in the latest trial. So what can we do next? Oh yeah, before I forget, I also would like to show this example to you. This is a study that was performed in Nijmegen by Bas Tunneberg et al. And he investigated the compound mexiletine that is used to treat myotonia, and he tested whether this also worked for non-dystrophic myotonia. And the reason why I want to show this is also because of the combination of the previous talks, where actually one of the challenges in rare disease patient research is the low numbers, for example, of patients that are available to do trials on. And also sometimes the large burden that these patients perceive by participating in such a trial. In this trial, we tackled that in two ways. So first of all, we had a crossover design of people being on and off the drug for some period, and that interchangeably. And that helps to actually reduce the number of patients that you need in a trial. And the other thing we tackled by not having the patients come to the clinic all the time, but actually we had an interactive voice response diary. And so regular, almost daily reports of self-reported stiffness. And actually what you see here in the graph is that almost all the patients saw a reduction in this muscle stiffness in the periods that they received the drug. So drug repurposing often initiates in academia. That's first of all because industry is not so much interested in drug repurposing because they earn a lot more money on novel chemical entities. And also because academia feels, I think, a special responsibility to do something for patients living with rare diseases, whereas many companies still focus on the common diseases. So in this recent review in Europe, 13 of the 68 approved orphan medicinal products were actually repurposed drugs, and 75% of these drugs and their development started in academia. So then we also embarked on this endeavor with the Sympathetic project. And in Sympathetic project is a consortium behind, including, as I said, the CHIO Research Institute in Ottawa, but the consortium consists of basic researchers, clinical researchers from different universities across Europe and Canada, by patient with patient organizations, and also with companies. And we feel that really the development of such a drug repurposing project is a multi-stakeholder effort where all of these parties need to be represented. Sympathetic's main paradigm is that we would like to have larger group of rare disease patients benefit from repurposed drugs by leaving behind the one drug, one disease paradigm, but developing drugs in parallel for different rare diseases by targeting overlapping clinical symptoms and the underlying pathophysiological mechanisms. So we chose 9 disorders, including myotonic dystrophy, but 9 disorders that are somewhere in this triangle between neurometabolic disorders, neurological disorders, and neuromuscular disorders. These disorders are actually sometimes difficult to classify, but they share some clinical symptoms like fatigue, movement disorders, and cognitive impairment. And we think that these clinical symptoms can also partly be represented in cultured cells, induced pluripotent stem cell-derived neurons that we profile for mitochondrial dysfunction and altered synaptic transmission. And these will be the cells that we do our drug screens on in parallel for all of these 9 disorders, and that we select candidate drugs from. So the acceleration in the drug repurposing pathway is then manifold. But in this project, it starts with the co-creation process with multiple stakeholders. So the patients, the clinical researchers, the basic researchers, and industry. The start is actually the patients and their symptoms and the classification of their symptoms in patient registries. Then we make in a standardized protocol induced pluripotent stem cell-derived neurons from the subgroups of these patients. We do a AI-empowered drug screen and actually then start to test these drugs that show efficacy in this neuronal model immediately in a trial that can include patients with different disorders. We call that a basket trial. And this leads to blueprints for exploitation of these drugs and regulatory approval. So the way how we would like to accelerate drug repurposing is several-fold, and I will spend one slide on each of these accelerators. So to integrate the patient perspectives and priorities in all the relevant steps of the drug repurposing pathway, to train patient advocates to drive the drug repurposing pathway themselves. And we see that patient advocates can really help to advance a drug repurposing study, to have dedicated preclinical models and readouts available for the rare disorders, to do then both the in vitro work, so the drug screening in parallel for many different rare disorders, and to also do the clinical development in parallel, testing in clinical trials patients with different disorders. And in the end, to deliver reusable exploitation and patient access model to the community. One of the tools that we are developing is the target product profile that actually helps you to, in a multi-stakeholder co-creation process, to go from what is needed to bring the drug on the market all the way back to actually how you would design your experiments. And this is typically what a target product profile looks like. It's nothing more than an Excel file that contains all the important aspects of a drug, both from a technical perspective, but also from a patient or user perspective. Things like disease indication, drug substance, drug product, clinical safety and efficacy, and drug access mechanisms are all listed in this Excel file, which is then used also as a means to discuss the co-creation process with all the stakeholders. As I said, one of the unique parts of our project is that we do the drug screens in relevant cell types, in this case in neurons, and that we have a very fast protocol to induce from a stem cell the neurons in about 7 days, and the success of that is shown in the stainings over here. Then we have standardized protocols for that, and we have also standardized imaging-based assays that measure mitochondrial activity and that measure synapse formation and neural signaling between neurons. And we are currently adapting that for a high throughput screen. Then about the trial that we are developing. The trial in this case also has a crossover design, very much like the Maxillitin trial that I showed you before. So where patients are partly on the drug and followed by a washout period and partly off the drug. That really helps to reduce the number of patients recruited to every sub-study. And we use the same protocols to test patients in different groups, different sub-studies, based on the different disease groups that we identify and that are showing a positive response to the drug. So the drug is always the same, but the patient may actually differ between the different sub-studies. And that is another increase in efficiency of this type of studies. Then we know that the variability between patients is huge, in particular because we actually include patients with different disorders. And our possible solution to that problem is to use goal attainment scaling as an outcome measure. And goal attainment scaling is it can be done across different disorders because the goals of that should be achieved after the therapy are personalized and also defined by patients themselves in dialogue with their healthcare providers. So the choice of goals is personalized. The scaling is the same, where 0 actually is the intended effect of the drug. For example, I can walk the stairs again, and 2 is then much better than expected, and -2 is much less than expected. And we compare at this goal attainment scaling also in a setting, in a crossover setting, so you can actually compare the same patient, whether he does better on the drug than on the placebo. Well, of course, there are still challenges in accelerating this drug repurposing pathway. much of the incentives for successful commercialization lie in new intellectual property. However, in drug repurposing, it's much more difficult to obtain relevant patents. So the market may not be big enough in particular for ultra rare diseases, even if the drugs are much cheaper than like gene or genetic therapies. Sometimes new pharmacodynamic data may still be needed to be generated because the original data that was used for the approval of the drug for another indication may have been on a different target group, for example. And what we also have seen is that the new registration of a drug by a commercial entity may actually lead to an enormous increase in the pricing of the drug for that new indication. And of course, that's undesirable. So some solutions in this area are the orphan drug designation status that provide a few years of market exclusivity for drugs treating patients with rare disorders. We now see both in the EMA and FDA schemas that really make the reductions in the regulatory pathway, fee reductions for academic partners. And we see the developing of new price models. And my last slides are on that. So here again, this is the example of mexiletine for non-dystrophic myotonia that I showed you earlier. When actually the company brought this drug to the market for the new indication, the price was enormously high. So the list price of this drug for a yearly equivalent was 900 times larger than the original list price. And this was actually a study where they actually calculated what would be a reasonable price for this drug, also taking into account the additional efforts that a company needed to do for the registration of this drug. And we refer to that as value-based and cost versus value-based pricing. So this is cost-based pricing, and there are alternatives as well, like performance-based pricing. So actually reimbursing based on how well the drug treats the symptoms. When you want to learn more about drug repurposing, I really need to refer you to the ERDIC Drug Repurposing Guidebook. The ERDIC is the International Rare Disease Consortium, and they have the Orphan Drug Guide and also special sections about drug repurposing, all aspects of drug repurposing, going from the stakeholder mapping to collecting relevant resources and information about the disease, defining the target product profiles and also the target patient value profiles, all summarized in an easy to navigate building blocks. So wherever and which step of the drug repurposing process you need help, this may be a first good entry of information also for patient advocates who would like to start a new drug repurposing project. Then in this paper down here, we list all kinds of initiatives that help actually the drug repurposing forward, because drug repurposing is currently, I think, a hot area of research and development in which many initiatives, including regulatory initiatives, are stimulating drug repurposing projects. Key to successful drug repurposing is, I think, engaging different stakeholders, including patient advocacy organizations in the process. And we hope to further increase the drug repurposing for rare diseases by leaving the one drug, one disease paradigm, but running drug repurposing pathways for multiple disorders in parallel and attacking some of the challenges in the commercialization and regulatory approval, but they will probably be there to stay for quite some years. This was my presentation. Thank you very much for your attention. Thank you very much to the funding and also the additional funding that the Canadian New Frontiers in Research Fund provided to this exciting project. And I'm happy to take questions.
Thank you, Dr. Tone. That was wonderful. It's also something Dr. Burney mentioned. He's interested in this topic, and so am I. I have a quick anecdote to tell you related to this. I was doing I am doing an RCT using an SGLT2 inhibitor, dapagliflozin, and want to study it, in a patient population where it's not currently indicated. And I approached probably 7 or 8 generic companies who now make this drug. It's fallen off patent. And I couldn't convince a single one of them to partner with me to do this project. Even though I have received funding from a peer review agency. So I think it is challenging to get, and it's even more challenging in rare diseases just because it's, fewer people affected and they may not see profitability from that. So you mentioned that you have some partnership with industry in sympathetic. So can you tell us a little bit about that? Are these generic companies and how is that working exactly?
Yes, so we are also talking to generic companies and have the same difficulty convincing them to embark on this kind of projects. Well, the jury is still out. I'm not sure whether we will be successful. But in the Netherlands, yeah, there is currently a trend to really keep everything in the own hands of academia, including also the regulatory pathways, so to really develop that expertise in-house with all the university medical centers in the Netherlands. So that we are less dependent on willing companies actually to actually put the drug on registration.
Maybe I can ask Peter. What's the role of animal models in this whole repurposing pathway? You mentioned at one time. Is it mandatory? Many of rare diseases don't have an animal model. Can you go straight to the stem cell stage, or is the stem cell stage just replacing animal models?
Yeah, it's a very good question. So indeed, we are trying to make a direct step from the induced pluripotent stem cells of patients to the clinical trial without doing first animal research. When there is a good animal model available, I think the regulatory authorities still favor that you would do also experiments in animals, but they also are very aware that the animal model may either not exist or have a very bad representation of the human phenotype. And in that case, I think there are quite some examples now where a drug, in particular, when its safety is well proven and the benefit risk ratio is good enough to directly to go to the testing in patients without first proving the efficacy in animal models. The animal models are, I think, still essential for toxicity studies, but yeah, usually these studies for repurposed drugs have been done in the past, and that data is typically available.
Okay, thank you.
Are you aware of any drug repurposing work in any cardiac disorders? Or I think a lot of the work you spoke about was neurologic or other conditions. So are there any rare cardiac conditions that you're aware of or we're studying repurposing drugs?
Yeah, so I'm most aware of a study in Nijmegen actually that was on a cardiac condition, but not a rare cardiac condition, and that was actually a story on that was not so well it was very successful, but too successful. So a company basically took over and put the drug on the market, even without involving the researchers that performed the clinical trial, and actually also at a high price. So I think it was an example of how we shouldn't do it. But there may be other studies also in rare cardiac conditions, but I don't have the overview of that.
Ian, I think you're peripherally involved in the target study in the RVC. That's repurposing a drug from somewhere, isn't it?
Yeah.
Yeah, I actually don't know which medication it is repurposing. Can you remember?
I have to look up my email.
Yeah, anyway, there's a study in Canada, phase 3 study run from PHRI McMaster, repurposing a drug for arrhythmogenic right ventricular cardiomyopathy, which is a rare cardiac.
Okay, well, I think we're at time. Thank you very much, Dr. Tahone. It's a topic that I really think is important in rare diseases, but also even across medicine. Just the cost of doing clinical trials for new drugs is really out of hand. And if we can find equivalent therapies that are much cheaper to study and get answers more quickly, everyone benefits from that. So thank you very much for your work on this, and great to see that you're working with Ottawa and Canada on these initiatives as well. So I'll introduce our last speaker for the morning session. It's Dr. Andrew Mackey. So Dr. Andrew Mackey is a professor and pediatric cardiologist in the Faculty of Medicine at the University of Alberta. He works at the Stollery Children's Hospital. And he studies challenges facing adolescent patients transitioning to adult healthcare, including lack of trust of new providers, difficulty of teenagers to manage their own illnesses. And these can obviously result in care gaps and negative health outcomes. And we obviously thought this is very relevant to our conference today because there's such a high genetic component to rare cardiac conditions. Many patients will be diagnosed in childhood and require transition to adult care. So today he's going to be talking about transition of care from pediatric to the adult setting. With that, thank you for joining us, Andrew.
Morning, Ian, and everyone. Thanks very much for having me. I'm flattered to be here, and there have been some fantastic presentations that preceded this one. And I'm just gonna, my slide doesn't want to navigate forward, so let's just try that again. There we go. I don't have any disclosures. My objectives are threefold. First of all, I hope that by the end of this presentation, you'll be able to describe the challenges encountered by transition-age youth, summarize key concepts of transition preparation, and identify at least one way of improving transition-related care in your clinical practice. And here's how the talk is going to be structured. I'll begin by describing some of the gaps in transition, going through what evidence we have right now in terms of how to improve transition, identify where there are knowledge gaps, and hopefully leave you with some practical tips. And like you said, Ian, many of our cardiac rare disorders have a phenotype that begins in childhood, sometimes even prenatally. And many of these children and adolescents have really had a very long and complex medical journey through the pediatric system. And setting them up for success as they enter adult healthcare is really paramount for their long-term well-being. So what is transition, and how does that differ from transfer, which is another term that is sometimes used synonymously, but they're not the same. So transition is a process. It's the purposeful, planned preparation of adolescents and young adults to succeed in the adult healthcare system. And if done well, fosters developmentally appropriate skill acquisition over time. It's not a single event in time. It typically takes years, and it spans, importantly, both the pediatric and the adult healthcare systems. On the other hand, transfer is an event. It's when the responsibility for care moves to the adult clinic, and it's part of transition, but just a small part. And to illustrate this, on the y-axis, I've put the term self-management, which I will describe on the next slide. And on the x-axis, we have age from early adolescence through to about 25 years of age. And you can see that self-management skills increase episodically, and that there are some plateaus, but they continue to increase beyond the age of 18. And in Canada, the typical age of transfer is about 18 years of age. So self-management is a set of methods, skills, and strategies by which individuals with a chronic health condition can effectively take care of themselves, in partnership, of course, with their healthcare team. And so, for example, this might include tasks like managing their medications or checking their blood pressure at home. And in order to self-manage, this requires, of course, some knowledge of the health condition, some problem-solving skills, access to and confidence in supports, and knowing where and when to turn for help, and emotional well-being. You can imagine that with mental health challenges, managing your health condition, your cardiac condition, can be additionally challenging. So I'd like to hinge this on a case. JR is a 20-year-old female with a history of idiopathic dilated cardiomyopathy that was diagnosed at age 12. With declining LV function gradually during adolescence, and her latest EF was 20% when she was seen in the pediatric cardiology clinic 18 months ago. Of course, it wasn't meant to be 18 months between the final pediatric appointment and the first adult appointment, but she moved out of her parents' home and she can't be reached any longer by the new healthcare team. She stopped taking all of her meds because she felt okay until she didn't feel okay, showing up in a walking clinic with breathlessness and fatigue, and was treated for community-acquired pneumonia, and 2 days later ends up in emerge in cardiogenic shock. So cases like this happen, and we do experience deaths during transition age years, and this is an entirely preventable situation. Young adult survivors are on the move. They're undergoing multiple other life transitions at the same time, and these include changes in school, new jobs, new partners, and other distractions. And it's no surprise that going to medical appointments is hardly priority number one. And this is a period of changing influences where family and healthcare providers become really much smaller influences relative to peers and social media. And psychologists have termed the age of 18 to 25 years as emerging adulthood, and this is a unique developmental period during which neuroanatomic and cognitive changes are still occurring. And anyone who's got teenagers at home, or anyone who's ever been a teenager themselves, can probably relate to some aspects of this cartoon. But seriously speaking, a chronic health condition may result in additional challenges above and beyond being a healthy young adult. For example, frequent hospitalizations and medical appointments can result in school absences, some disconnection from peers, higher incidence of anxiety and depression, and difficulty developing an identity outside of the illness. And all of these complicate the ability to manage the chronic illness. And as a result, transition can be a period of high medical and psychosocial risk. So the consequences of transition include healthcare dropout, difficulty attaching emotionally to new adult providers, poor treatment adherence, increased emergency room visits, and increased hospitalizations. And this has been demonstrated not only in congenital heart disease, which I've studied the most, but also across a variety of other pediatric onset chronic conditions. And what are the impact of lapses in care for those with rare cardiac diseases? Well, I've oversimplified this a little bit, but here's some examples. So for those with cardiomyopathy, lapses in care can result in progression of heart failure symptoms. For those with Marfan syndrome or other genetic aortopathies, undetected aortic root dilation will be the consequences of lapses in care, which can result in preventable dissection. For those who've had a heart transplant, graft rejection, cardiogenic shock, and death can certainly occur from lapses in medication use and lapses in care. And for those with inherited arrhythmias, not receiving appropriate medical therapy or device therapy, and sudden death. Well, our young adults and parents have important perspectives that we as providers need to be aware about as it relates to transition. And these are generalizations, admittedly, but youth typically feel quite unprepared to manage their health. And in the absence of a transition program in pediatric care, they feel like they lack the basic self-management and self-advocacy skills. They don't know where to turn for help. And this is partly the explanation for increased emergency room visits in 18 to 21-year-olds. That's been documented in the province of Alberta. They feel relatively attached to pediatric providers, and they may have cognitive delay, which challenges all of this. Parents, on the other hand, really have difficulty letting go of the healthcare responsibilities related to their young adult child. More so than youth, they have very strong attachment to pediatric providers. They're quite wary of providers in the adult system, and of course, they remain very invested in the health of their young adult child. And more so than their youth, they experience a great deal of anxiety. And I've had some mothers tell me they're literally terrified of leaving pediatric care. Mental health conditions. So anxiety and depression are 2 to 3 times more common in adolescents with cardiac disease, and this increases difficulty with self-management. And in fact, among adolescent heart transplant recipients, having a prior mental health hospitalization strongly was associated with death during transition-related years. And this is a study from Philadelphia. So it's hardly surprising that entering the adult healthcare system can feel like jumping across a wide and deep chasm. And so what are we actually doing as healthcare providers to fix the transition problem? And the answer is certainly more than was being done 10 to 15 years ago, but certainly not enough. And a recent review of North American and pediatric cardiac centers demonstrated that still about 60% do not have a structured transition program for their graduating teens. Unfortunately, most teens and young adults with cardiac disease have quite limited knowledge of their heart, and this has been demonstrated repeatedly. But a really interesting study from Sick Kids a number of years ago demonstrated that teens who are knowledgeable about their cardiac disease are more likely to be confident communicating directly with their doctors or nurses rather than having their parents do so. And obviously being able to have a conversation with your doctor or nurse and knowing what questions to ask and what information to share is an important part of self-management. So this got me thinking that we need to focus on developing knowledge among our patients, and we didn't have an evidence base at the time. So I embarked on a clinical trial which had 2 aims. First, to develop and evaluate a clinic-based transition intervention for youth with congenital heart disease. And second, to measure CHD knowledge and self-management before and after the intervention. And we called this the chapter study. It enrolled 15 to 17-year-olds with moderate or complex CHD. And there were 2 groups. The intervention group worked with the nurse one-on-one for a teaching session in conjunction with a clinic visit. And that lasted about an hour. And the control group didn't receive any teaching. On this slide and the next slide, I'll summarize the elements of the intervention protocol. We have a participant on the right who agreed to have a photo taken, and one of our nurses. So they began by discussing transition and what that term means and what's going to happen over the next couple of years. They created a health passport together, a succinct summary of the young adults' health care conditions, including the names of their conditions, summary of previous casts and surgeries, the name, dose, and rationale for their meds. They reviewed a diagram of their anatomy and discussed potential late cardiac complications, which was the opportunity for the nurse to send the message that they have a lifelong condition and that they're not cured, which is a common misconception. The participant then received the names and location of the adult congenital heart disease clinic. There was a discussion of risk-taking behaviors using third-person scenarios. Some youth-friendly take-home written materials were provided in terms of the impact of sex, drugs, and rock and roll in general on their congenital heart disease. They were introduced to some patient-oriented websites, and then the nurse followed up with the participant within 7 days to see if they had additional questions or concerns. And what we found is that knowledge of their condition, which is on the y-axis, increased in the intervention group in green compared to the usual care group in red at 1 and 6 months. And we don't actually think that they, of course, memorized everything that they were taught, but having a health passport, which they could refer to at any time, was probably why their knowledge scores were still good at 6 months. We also looked at self-management scores using a validated questionnaire. And we were a bit surprised, but very pleased to see that in the intervention group, self-management scores were higher at 6 months. This is perhaps a reflection of the increased knowledge that they had about their condition. Then we followed this up with the Chapter 2 study, which asked the following questions. What's the role of a nurse-led one-on-one coaching session that's specifically focused on self-management? And does this intervention impact adult congenital heart disease clinic attendance? Because if you don't arrive in the adult clinic, well, then you're not going to successfully enter the adult healthcare system. So in the Chapter 2 study, there were 2 sessions. The first one was very similar to what I described with the Chapter 1 study. But in addition, we added a couple of other elements. There was a goal-setting exercise that the nurse created for the participant, and they visited the adult clinic. And many adolescents have a lot of anxiety simply about how they're going to get to their first appointment. They don't know where it is. And so that was simple for us because they were literally a 5-minute walk between the pediatric and adult clinics. The second session was held 2 months later, and the role of the nurse was not really that of a teacher, but more of a coach or facilitator. And the second session was focused on self-management skills, and it included a review of the goal-setting exercise, how to make an appointment, how to get ready for an appointment, figuring out what questions you have for your doctors and nurses, how to fill a prescription, and some role-playing scenarios. And what we found in the Chapter 2 study was that the probability of attending the first adult appointment on the y-axis was more timely in the intervention group in blue compared to the usual care group in red. And we were pleased to see this because lapses in care are well described during the transition age years in congenital heart disease. And in fact, here's a recent systematic review. So globally, there's discontinuity of cardiac follow-up or loss to follow-up of 2 or more years in about a quarter of patients. That's more common in the US, but in Canada, we're, a quarter of our congenital heart disease patients are lost to follow-up, even in the current era. In the European Union, they're doing a lot better, and so that means that, there's definitely room for improvement in North America. This is the percent to loss to follow-up for simple, moderate, and complex congenital heart disease, and it's sobering to see that more than 1 in 5 of our patients with complex congenital heart disease have lapses in care. And this systematic review, which included the Chapter 2 study I just described, demonstrated that programs having clinical centers having a transition program are associated with a much lower rate of loss to follow-up among their patients compared to centers not having a transition program. What I would say we really learned from the Chapter studies is that you need dedicated nursing time to do this well. You need some space within the clinic area, which can be hard to come by. Teaching interventions need to be on the same day as the clinic visit. Parents struggled with being left out related to the anxiety they experience about all of this. And health passports need to be smartphone friendly, and we've made it that way now. So just turning this information back to rare cardiac diseases, there's a paper out of Philadelphia a number of years ago that obtained information from patients and parents of young people with Marfan syndrome. And they told those researchers what information they desired during the transition age years. They want to know about the impact of Marfan syndrome on lifestyle and career choices. They want to understand better the long-term effects of Marfan syndrome. They want to understand the purpose, expected effects, and side effects of their medications. They want to have a better understanding of how to monitor themselves in terms of heart rate, blood pressure, and symptoms. They want a better understanding of terminology, both medical and lay alternative. And they really want a better understanding of treatment options that they face in the next few years as they enter adulthood. And so this is basically an education curriculum that aortopathy clinics could certainly easily address. What about patient navigation? There are a variety of different terms that have been used to explain these job titles, including navigator or transition coordinator. In my experience, and I've had the good fortune of working with one, I would say that a background in social work is extremely valuable. And navigators have the ability to provide support in multiple domains. Not only can they work with other members of the healthcare team to increase the self-management skills of young adults, but they are there to help young adults navigate the complexities of the medical system, especially when you need to not only see a cardiologist, but you need to see an ophthalmologist and an orthopedic surgeon for your Marfan syndrome and a medical geneticist. It can become overwhelming. And navigators can help address and mitigate socioeconomic barriers that may prevent patients from accessing care. And in the province of Alberta, I've had the good fortune of co-leading with some colleagues in Calgary the Transition Navigator Trial, which is a pragmatic RCT of 16 to 21-year-olds living with a chronic health condition. The intervention group worked with a navigator who, again, was a social worker for 12 to 24 months. Our primary outcome was ER visit rates, and the study was conducted across the province. And there are some unique aspects of the role that I want to point out. So the navigator really took a whole person approach. They didn't just think of the person as a person with a rare cardiac disease or as a person with congenital heart disease. They set them up with links to the community in terms of education. Some of our participants had unstable housing, and the navigator was able to help with that. Mental health and addiction and financial assistance were necessary. And we've published a couple of papers this year. We had over 300 patients. The primary outcome was ER visit rates, and we observed an effect modification in that there was fewer emergency room visits in the intervention group for those with a mental health condition. Although among the overall cohort, the navigation did not decrease the number of emergency room visits. The real benefit of the navigator was evident through our qualitative research, and three themes came out: emotional support, information and task-focused support, and guidance to become more independent. And some quotes provided from participants are there on the screen. And then I'd like to also identify really interesting body of work that's been led by colleagues of mine at Sick Kids. This is the iPeer-to-Peer program, and this is a publication of heart transplant and lung transplant recipients who had a virtual peer mentoring program. They enrolled 20 mentees, aged 12 to 17, and 9 mentors, aged 18 to 25. And the mentors were transplant recipients who had successfully transitioned to adult care already. And the mentees and mentors met one-on-one every 1 to 3 weeks over a 15-week period. The calls were recorded and subsequently listened to by a member of the Sick Kids team. And the median call length was about 22 minutes. And the mentees benefited in terms of increasing their self-management skills and fewer symptoms of depression and anxiety. And the mentors also benefited in a number of ways as well. And I thought this was really fascinating work, and there's more to come. So I've described some potential transition interventions: education about the condition, self-management skills, patient navigation, peer mentoring. But there are a number of other areas in which one could imagine we could improve the transition process, for which we don't really have data yet. I'm just going to mention one of them, technology and smartphone apps. At the moment, I'm leading the Ready or Not study, which is a randomized clinical trial comparing a transition app versus nurse-based teaching for adolescents with congenital heart disease. The app is quite lengthy. It's comprised of 20 modules, and each one takes 10 to 15 minutes. It's being conducted at 4 Canadian pediatric hospitals, and we've enrolled over 200 patients, and data collection's ongoing until the fall of this year. So more to come. And the final part of my talk, I want to leave you with some practical tips about transition. So a few thoughts about working with young adults. First of all, it's much simpler to coordinate education with routine clinic appointments, and this can be done for in-person appointments, of course, as I've described with the chapter studies, but it can also be done virtually. And the nurses I work with have really established an excellent way of doing this over Zoom. You need to progress at the individual pace of the young adult. Some of them will have challenges with executive functioning and cognition. You need to emphasize the need for lifelong care. And of course, you need to accommodate for low health literacy and or deficits in executive functioning. With respect to working with parents, yes, some parents are helicopter parents, but ignoring them is really not the right strategy. You need to explain to them the rationale of fostering their young adult child's independence if they're struggling to do that. And please inquire about their concerns and take advantage of their insights because they know their young adult child better than anyone else. And if your center doesn't have a transition program, then consider creating one. You need a multidisciplinary team with doctors on the pediatric and adult side, nurses, and ideally, if you can get it, a social worker and a psychologist. And having a transition program really opens the lines of communication between the pediatric and the adult teams. And these lines of communication are essential in terms of the transfer process. So that's when we pass the baton, pass the responsibility of medical care from peds to adults. And so transfer, points that I want to emphasize are, first of all, developing a strategy to track young adults who've been referred from pediatrics is really essential so you don't lose sight of them. You want to track the time intervals between the final pediatric appointment and the first adult appointment to make sure that you're not leaving these young adults waiting too long. As pediatric colleagues, we really appreciate being informed of the transfer outcome and consider avoiding dramatic management changes at the first adult visit. It's going to take a few visits for these young adults and their families to gain your trust. And making dramatic management changes right at the beginning can undermine that. So I'm almost done now. In summary, what we're aiming for is all patients arriving in the adult clinic in a timely manner with knowledge of their condition, a health passport on their phone, and some self-management and self-advocacy skills. We want patients to feel engaged with their adult team providers and want to return to the adult clinic. And of course, we want equitable access to care. So adverse social determinants of health need to be addressed to mitigate barriers to access to care. And this is where social workers are key. And ideally, we'd have the time, space, and human resources to do all of the above. And I realize that this is not easy, but it's worth striving for. I'd like you to consider this. What a missed opportunity it would be, having invested so much in these young people throughout childhood, to not set them up for success in the adult healthcare system. Collectively, as providers, we have the responsibility to instill relevant knowledge and skills in young adults that will set them up for success in managing their healthcare. I'd like to acknowledge my collaborators in the Transition Navigator trial, the chapter studies, and the Ready or Not CHD study. And I'd like to thank you for your time. If you have any questions or comments that you want me to email me about, please feel free to do so, and I've provided some resources there.
Thank you, Andrew. That was a wonderful talk. It brought up a couple of questions for me, but yeah, and I don't see. Yeah, oh, there's a couple of questions in the audience too. Maybe before I get to those, a quick question. Any thoughts about why Europe is doing better than Canada or US in terms of transition of care? So what is it specifically that they do that sees better performance of transition of care?
So I would say most of the data from Europe comes from just a relatively few number of countries, and those countries are geographically quite small. And so the geographic challenges that we experience in Canada don't really exist because many of our patients in Canada live very far away from tertiary care centers. So that's one reason. Of course, many countries in the European Union have a universally funded healthcare system, like we do in Canada. But the absence of a universally funded healthcare system, I firmly believe, contributes to the challenges that our colleagues south of the border are experiencing. And then it's really finally a question of the collective will of European centers to collaborate on the pediatric and adult front together and create high-quality coordinated transition programs. And overall, I would say they've had better success there than we've had in Canada.
Okay, we've got a lot of questions in the chat, I'm noticing. Let's see here. Great talk. Transition support makes sense. Are there data available showing that a structured transition improves the long-term outcome of patients with chronic conditions? How can we justify these efforts that putting into transition programs when healthcare resources are limited? So is there data supporting these transition programs?
Well, I've tried to share some data in terms of the transition programs. I would so certainly having lapses in care for 2 or more years is significantly less likely to occur for patients who've had the opportunity of going through a transition program.
Any sort of cost effectiveness studies, Andrew, I guess?
No, I'm not aware of any cost effectiveness studies. No, that's an excellent question. I mean, there is data demonstrating the clinical impact in congenital heart disease of lapses in care, and so a study from Colorado, for example, demonstrated that following a lapse in care, between pediatric and adult, those who'd had a lapse in care were 3 times more likely to require an urgent cardiac intervention within 6 months of arriving into the adult clinic, relative to those who've not had a lapse in care. But in terms of a cost-effective analysis, no, I'm not aware of any.
Kristen Lyons makes a good point and question. So some patients have multidisciplinary care with many specialists, and maybe transition of care may happen good in one of these, may be effective in one of these specialists and not so much in the other. And it's something I see, with a disease, let's say Fabry disease, where you could have a cardiologist, nephrologist, a metabolic specialist, a genetic specialist. And many people work in silos, and there may not actually be an adult equivalent to transition the patient to, right? So any thoughts around that when there's, trying to transition patients with multidisciplinary care needs?
Yeah, absolutely. It sometimes on the pediatric side, we struggle with feeling confident that there are providers on the adult side who have the skill set and interest to manage some of these complicated patients. And that certainly does not apply in congenital heart disease, where in Canada we have excellent ACHD providers. Having a family doctor is really key as the quarterback, and we certainly do what we can to help our patients who don't have a family doctor, obtain one. And certainly some general pediatricians will hold on to their complex multi-system patients into their early to mid-20s sometimes. because they don't have an equivalent person on the adult side that they feel they can transfer their patient to. I would say that multidisciplinary clinics are starting to pop up on the adult side. To my knowledge, and people like yourself and Kristen Lyons would have a better understanding, but I know that the adult congenital heart disease team, for example, in Calgary, have established a multidisciplinary Fontan clinic for patients living with single ventricle congenital heart disease. And so that allows these patients to see a hepatologist and a nephrologist and a cardiologist all in the same half day, which is excellent patient care. And more pediatric programs have established multidisciplinary Fontan clinics, but to see our adult colleagues do that too is really heartening.
The family who are affected and all have that same variant, that was enough to reclassify this as a likely pathogenic. So this case illustrates two important concepts. The first and the most important one is that you could use these segregation analyses in a variant of uncertain significance to potentially reclassify either as likely pathogenic or the other direction. If you have a variant that it's not present in multiple people in the family who are affected, then that means that this is probably not the driver. The second important concept is that, again, there are some genes that can cause a different phenotype. Even the same variant in the same family can have different phenotypes. MYH7 is the most common gene to have this happen. There are not many genes where it can happen, but don't let a family where there's hypertrophic cardiomyopathy in one person and dilated in a different person make you think that maybe it's not. All right. So for the last few minutes here, I'd like to focus on neuromuscular disease. What I want to do is to be able to go through and have a little bit of time for questions at the end. And another disclaimer for neuromuscular disease, when I started 10 years ago in a genetics cardiology clinic, I was seeing a lot of neuromuscular patients. Over the years, my practice has gone more towards just the cardiomyopathies and the aortopathies. And one of our newer genetics cardiologists has been taking on more of the neuromuscular disease patients. I actually haven't seen a neuromuscular patient for a couple of years, but the take-home messages are ones that have really stuck with me. And I want to be able to share some of those highlights with you. I also want to thank Dr. Beth McNally. She is our preeminent neuromuscular genetics guru, and she has shared some of her slides with me to help with this talk. So the particular neuromuscular diseases to keep in mind as a cardiologist include the muscular dystrophies. Those are Duchenne and Becker muscular dystrophy for the DMD gene, and then limb-girdle muscular dystrophies, Lamin, EMD, several genes that can cause those different subtypes of limb-girdle muscular dystrophy. Second is myotonic dystrophies, type 1 and type 2. And then much smaller groups include Friedreich ataxia and Pompe mitochondrial myofibrillar diseases. I'm going to focus on these two groups for the rest of the talk here today. And some of the things to point out are that a genetic diagnosis is really critical for helping to define those cardiac risks. As I mentioned at the beginning, the particular cardiac conduction versus heart failure risks are different depending on what the genetic diagnosis is. Those risks include cardiomyopathy and heart failure, as well as arrhythmias. And it's important to know that some of the more conventional markers of heart failure may not apply. BNP can be helpful, but it may not reflect the amount of disease. Biomarkers are also very important, and that these individuals, not only do they have skeletal muscle disease, but often diaphragmatic disease. And so a multidisciplinary team with having your neurologists, cardiologists, pulmonologists, and social workers all together is very important for optimal management of these patients. For Duchenne muscular dystrophy, this is diagnosed usually around 5 years of age. This is an X-linked disorder. And so in most cases, it's males that are affected, but females can have a mild cardiomyopathy. So we always want to make sure to be screening and following the maternal carriers of the DMD mutation. For the males who are affected, there's loss of ambulation by age 13 in general. And for many years, this was a disease that was fatal in childhood or early adolescence, but steroids and ventilatory support have improved survival. And now what we're seeing is that heart failure is the most common cause of death. The cardiac MRI on the right with the late gadolinium image is showing that profound amount of fibrosis that does develop in the hearts of these young men with Duchenne's muscular dystrophy. Becker muscular dystrophy is less severe, but there is still generally a loss of ambulation by age 16. Now, the diaphragmatic disease is not quite as severe in Becker, and there are some Becker muscular dystrophy patients who have a heart-specific phenotype or a heart-predominant phenotype is what I should say. And we've had some cases where they've progressed to the point of needing a heart transplant. And if the other aspects of their skeletal and diaphragmatic disease are not as severe, it's actually the case that a Becker patient is not absolutely contraindicated from heart transplant. And then finally, there are cases in women, then the carrier moms, of having an isolated cardiac phenotype. Within the muscular dystrophy group, we have found that steroids not only prolong survival, it's that they are delaying that LV dysfunction onset. So this is a retrospective study where we had 291 patients that were treated with steroids and 171 that were not treated. On the x-axis is the patient's age and then their function. And you can see those that were not on the steroids were developing LV dysfunction by age 15, whereas those who were on steroids, that was delayed by a good 5 or more years. Furthermore, ACE inhibitors have been shown to improve survival. And so over this 10-year study, we can see that the use of an ACE inhibitor had a tremendous effect on survival as compared to those that were on placebo. And for these reasons, then we've set up kind of a basic care plan for patients for their cardiac care. That would include an echocardiogram starting in childhood, age 6 to 9, and then every year. EKG, same time. Holter monitors a little bit older, and then that can be every 1 to 3 years. And getting a baseline cardiac MRI is very important, and then repeating that every 3 to 5 years is the most common. GDMT should be used in individuals who are developing LV dysfunction, but hypotension can limit their use. Using the BMP can be helpful. A lot of times when we talk about heart failure symptoms, we use ambulation as that first clue, and these patients have already a baseline ambulatory defect. So BNP is important for screening. Creatinine is also important to know that their creatinine is going to be very low because their muscle mass is so low. So if you need to look at renal function and monitoring that while these patients are on their ACE inhibitors or ARBs, then you should use a serum cystatin C. Using troponin to understand the amount of myocardial damage is helpful. LFTs can be elevated, and then again, female carriers can be affected, so they should be followed. Now for the myotonic dystrophies, there are two types, type 1 and type 2. Type 1 is the more aggressive form. It's caused by CTG repeats in the DMPK gene. The type 2 is caused by CCTG repeats in the CMBP gene. And then the presentation with type 1, it's usually more distal muscle weakness more than proximal, whereas it's the opposite for type 2. They both have a grip myotomia where they grip down and then they can't release the grip. And so this is usually one of the very early features. Features also include these Christmas tree cataracts. And then the facial skeletal features are that there's frontal balding, temporal wasting and ptosis is often seen. And then in type 1 in particular, this distal wasting of the muscles. There are extramuscular manifestations to keep in mind, so cataracts, diabetes. It's very, very common having GI symptoms and hypogonadism. But then the cardiac manifestations that we need to be aware of are the atrial arrhythmias, having cardiac conduction disease, ventricular arrhythmias, and then cardiomyopathy with heart failure. And what I want to spend a moment on are our guidelines for devices in these patients. So for myotonic dystrophy, the class 1 indications for bradycardias and conduction arrhythmias. In anyone who has a low EF, then they should be getting an ICD that has pacemaking capability. Anyone with symptomatic bradycardia, like our usual guidelines, then a pacemaker. And that even if it is not symptomatic yet, but they're in third-degree heart block, then of course getting a pacemaker in these patients. But what I want to point out is that there are two-way indications from the Heart Rhythm Society guidelines that if there's even just marked first-degree or second-degree AV block, that it's reasonable to consider a pacemaker in myotonic dystrophies, and that it's reasonable to use an EP study to try to identify the risks for high-grade conduction block early. For ventricular arrhythmias, then anyone who meets guidelines for a defibrillator with myotonic dystrophy, be sure to use a defibrillator where you've got pacemaker capabilities as well. And so that means, of course, a transvenous device. The subcutaneous devices where you're not able to do atrial pacing are not going to be appropriate in this population. And then the final one to keep in mind is, as a 2B indication, that anyone with type 1 or type 2 myotonic dystrophy who is getting a pacemaker, it's reasonable to consider going straight to a defibrillator depending on what their clinical presentation is. So if they have a lot of ventricular ectopy, even if there's no non-sustained VT yet, or if the ejection fraction is starting to go down, it is reasonable to consider an ICD at that stage. And so just to summarize the take-home messages here for implementing genetic testing in practice, that if you have a patient with dilated cardiomyopathy, neuromuscular disease, anything where you're considering, should I get a genetic diagnosis, referring for the genetics clinic where a 3-generation family history can be obtained, providing that pretest genetic counseling where you talk about what those types of results could be so that when they receive their results, they have that background knowledge already, sending off the panel-based testing, and then bringing the patient back for a results discussion and additional genetic counseling where we would counsel for patients who have a pathogenic variant. We're able to provide medical management advice to the referring cardiologists and then to initiate the testing of family members. For those with benign variants, then to be able to do clinical screening, because the absence of a genetic variant does not mean that there's no genetic risk. And that for variants of uncertain significance, to continue screening the first-degree relatives, but then also using the testing of affected family members for segregation studies to potentially reclassify that variant. And that is all for here. Thank you very much. And I'm happy to use the last few minutes that we have for questions.
Thank you so much, Dr. Wilsbacher. That was a fantastic overview. We have one question so far in the chat from Noor, who's asking if there is a certain number of people in a family that need to be affected to reclassify a VUS as likely pathogenic. So using that case example you gave, for instance, and ultimately what processes are involved in reclassifying a VUS within the center.
Excellent question. So the number that we most frequently will invoke is 5. And if you have 5 family members across 3 generations, then it's going to create a LOD score that's high enough to cross that threshold for reclassification. We do this working with the genetic testing companies because part of variant classification is being able to distribute that information to the rest of the field. There are resources, including a site called ClinVar, where all of the genetic testing companies around the world will input the variants that they have observed and their classifications. And in many cases, they'll even give some of their rationale behind that classification. And so when we reach a point working with a genetic testing company that we now have enough at the genetic testing company, everyone agrees that this is now likely pathogenic, then that also gets upgraded in ClinVar, and other investigators who may use different genetic testing companies may start to see that. There's another consortium called ClinGen. It is a collection of experts, genetic counseling and clinical experts, PhD scientists, who convene councils essentially to review genes for likelihood of pathogenicity and to even review particular variants to assess whether that variant should be called likely pathogenic or not. And so the information that gets moved to ClinVar. And so anytime I see a variant that there is documentation in ClinVar that this is a adjudicated variant in ClinGen, and I know that it's the highest level of evidence that we have. Now, that being said, sometimes we get to the point where we are convinced that this is what's causing disease in the patient and their family. And for reasons that are complex, we don't get agreement from the genetic testing company that we've used. We tell these patients we're treating you as you have the disease, and we are going to do everything to continue management based on calling this likely pathogenic for your clinical care.
Dr. Wilsbacher, thank you. That's a phenomenal talk. You touched on the lack of diversity amongst genetic ancestry in the resources we use to classify pathogenicity of variants. And can you speak to how that lack of diversity would negatively impact an individual of an underrepresented minority who undergoes genetic testing, or what that might do to the test results? I hope that's not too opaque of a question there for.
Yeah. You know, it's one that it is a very, it's such an important question. And to give a little bit of background of how it impacted when we first learned of the amount of genetic diversity, a lot of variants had been classified as disease-causing in a European ancestry, and then were noted to be observed at much higher frequency in other ancestries, and that particular variant was not associated with disease. Before we had the larger amount of genetic data from the other ancestries, we had individuals of the non-European ancestry who had, they had basically been given a diagnosis and even received devices or received procedures that they did not need because they did not have a condition. And it goes back in the other direction within the European ancestry patients as well. In terms of whether there might be an allele that in a non-European ancestry is quite rare and could be disease causing, we still just don't have enough information to be able to unravel all of that. And the need for additional genetic studies in all ancestries is critically important so that we can use that information appropriately. This is also very important for the development of polygenic risk scores. As I'm sure probably all of you have been seeing in the journals, there are polygenic risk scores that are being reported frequently, but the data are based on whole genome and GWAS studies that are, again, primarily from European ancestries. And is it appropriate or possible to apply a polygenic risk score to a person of a separate ancestry? Probably not. And so this is one of the reasons that polygenic risk scores are likely not going to be widely clinically available until we have sufficient whole genome studies and GWAS data from other ancestries.
Thank you.
We got a question from Sonu in the group, a little bit of more purple pride. What is the difference from a clinical perspective between a likely pathogenic versus a pathogenic variant?
Yeah, that's a great question. Clinically, there's no difference. From a gene variant classification standpoint, likely pathogenic means that it is 95% likely to be disease causing. Likely pathogenic means that it's 90%. So we've got pathogenic is 95%, likely pathogenic is 90%. But from a clinical practice standpoint, we treat them the same. We treat them as a disease causing, and that family members can and should be tested for those variants.
Thank you. And I had a little bit also of a sort of ethical question. Thank you for touching on the intricacies of having, a law in place, the Genetic Non-Discrimination Act. Sometimes I get the patients asking questions like, how is that information coded, available? Where is it stored? There's lots of fear about privacy of data in the global sphere. And how do you sort of reassure your patients from that standpoint when they ask a question.
Yeah, absolutely. And we get that question all the time. The companies that we use, most of them will, first of all, discard the DNA after a certain amount of time. So 30 days, I'm sorry, 90 days is one common for one of the companies that we use, 1 to 2 years for another one. So the actual DNA material is discarded. The information itself, it is protected in the United States by HIPAA laws, and that they cannot be obtained without the consent of a patient. So this is unlike the ancestry testing where this is a direct-to-consumer testing, and part of the signature is the fine print is I'm allowing it to be used by other groups. So for anybody who does direct-to-consumer testing, you have to really be careful with that fine print. But with clinical testing, just like anything else in the medical records, it cannot be released to other parties without the patient's consent. Now, in terms of security of data from a more institutional standpoint, there's the same risks as any hospital, any university, any company would have of being hacked, that all of these companies take this very seriously and do everything in their power to protect that data that is there. But that is one aspect that we need to be very transparent with patients about, that we can't be 100% protected, because nothing can be 100% protected.
I think just to add to that some local context, I think we should be really proud in Canada, the Genetic Non-Discrimination Act, as Dr. Wilsbacher mentioned, was first codified in law in 2017. It was upheld in the Supreme Court in 2020. And it goes so far as to state that even if an insurance company were to obtain this information somehow, if it were to be left on their desk, that they can't actually use that. And we should clarify that there's a difference in these laws, the protection they provide to patients who are undergoing diagnostic testing versus predictive testing, right? So if you're undergoing diagnostic genetic testing, you already have a dilated or non-dilated LV cardiomyopathy. And this is something that you would have to disclose on a questionnaire when applying for insurance. And so where it's really of value is that once someone has a genetic diagnosis and you're offering predictive testing to family members, that they can feel confident that they can go have testing, determine if they might be at risk of cardiomyopathy in the future without that negatively impacting their insurability or employability. And so that distinction is really important. Now, insurance companies could ask, do you have a first-degree relative with cardiomyopathy? Right? And so there's ways that insurance companies could probe, but in terms of the genetic results, that is protected.
Couldn't agree more. And that was so elegantly stated. And thank you. It's one of the things that we spend the most time with our genetic counseling on, that life insurance piece, that the predictive genetic testing it's something that is at risk for life insurance in the United States. And like many things right now, I look to Canada as such a source of good things that happen.
I think we may have time, Dr. Music, for one more question of Dr. Wilsbacher here. This is from Dr. Swigum in Victoria. If a family history of a non-ischemic cardiomyopathy is present, but medical genetic testing is negative, I think in the proband, when should genetics be repeated on the proband?
Absolutely. We usually recommend every 5 years. The panels are adding new genes all the time. The likelihood of a test coming up with a new gene that becomes diagnostic in 5 years, it's still not incredibly high, but that interval of testing is cost effective and in some cases can identify a newly discovered gene that's important for cardiomyopathy. So every 5 years.
Yeah, I'll add to that. Recently at Ottawa, the panel has expanded from 30-some genes to 100. Our local panel was quite limited, so anybody who has a high suspicion should probably be retested with this new panel.
Okay, well, with that, we'd like to thank you, Dr. Wilsbacher, for an incredible talk and educating us. I really enjoyed that. And as we move on to the next half of the session, It's a great pleasure of mine to introduce an old friend and colleague, Dr. Thomas Roston. Thomas is a cardiologist and clinician scientist at UBC, and I first met Thomas when we were in medical school together, and I've kind of just been in awe of him ever since then. We went through residency training together, and he concurrently completed a master's. And then we went through cardiology training together, and he completed concurrently a PhD from the University of Alberta in translational medicine. He went on to do postdoctoral studies, including a fellowship in cardiovascular genetics at Harvard, and then a fellowship in cardiac critical care at the University of Alberta. He's a very active clinician scientist focused on the lives of young patients and their families who are at risk of sudden death or have suffered sudden cardiac arrest. He holds research grants from the CCS and was just awarded a very large project grant from CIHR to study genetic cardiomyopathy. And I've always admired Thomas for being such a principled and clear thinker. And I'm sure through this talk you'll see why he's really one of the rising thought leaders in genetic cardiomyopathy. So welcome, Thomas, and thank you for joining us.
Yeah, thanks so much for having me, Omid, and to Dr. Music and Dr. Wilsbacher. It's a pleasure to present in this session and to follow such a distinguished speaker as well. I was given the task of talking about the genetics of non-ischemic cardiomyopathy, which I think honestly nowadays you could probably give an entire graduate level course on this, but I'll try to distill it down into something that, is easy to understand in the next half hour or so. And also kind of explain for more of us who have, are general cardiologists, what is important to know and to understand to recognize these conditions. So as I said, it's a real pleasure to be here, and I look forward to your questions at the end. And we had a great discussion, I thought, at the end of that last session. So I do look forward to that as well. So the objectives of this talk, the first one was to review some basic genetic concepts, but I think our previous session really did an excellent job of that. So I'll just highlight a few key points and not reiterate everything that was discussed. And then I want to talk about the indications and rationale for genetic testing in non-ischemic cardiomyopathy. And then the last thing is to discuss variability and adaptations in nomenclature and classifications over time and how we think about cardiomyopathies in the modern era. And I have no relevant disclosures to this talk. This is a book that I used to really enjoy reading when I was a kid. It's called What a Mess. It's about a dog that's a complete mess and, turns the house upside down. And sometimes I think about non-ischemic cardiomyopathy this way because I find very few things are completely straightforward, and we're constantly changing our opinion about which genes are pathogenic, which genes are not. It really is a field that, and same with families, which person may be affected, which one maybe not. So I'll highlight some of these points, but I think it's an area that's really a wonderful place to do research because there are so many simple unanswered questions that still exist in this field. So the first thing, that I think is a bit of a mess is the exact classification and language we use around this condition, which is the presence of a weakened heart muscle not due to coronary artery disease. And, I will say that it's very uncommon in medicine that we use a diagnostic term that really is not a diagnosis. So we're really saying you have weakened heart muscle and we kind of just ruled out coronary artery disease. And then we realize from there's this massive differential diagnosis. So if you look at the guidelines and the position statements from the European Society, from American Heart, from Heart Rhythm, these amazing organizations that have incredible thought leaders leading these guidelines, you'll actually see some differences between them that are rather marked. And you also see over time that how we actually diagnose these conditions has changed. So if you're doing research in this field, what was considered DCM 15 years ago might actually not quite meet the same diagnostic criteria in 2014 and vice versa. So again, it can be really hard to follow the literature and understand, what truly is a non-ischemic cardiomyopathy and what diagnostic workup is necessary to reach that conclusion. So this is the Canadian Heart Failure Society guidelines, which I think is a great place to start just because it's a document that many of us have seen. And I want to point out two things in this guidelines. This is a nice figure that summarizes the assessment of someone presenting with de novo heart failure. And in two boxes at the end, it really emphasizes that if you have exhausted all other assessments of the patient and you still don't know why they have cardiomyopathy, you should be doing family history, you should be referring to genetics, and you should be considering some of these diagnoses in your patient. And surprisingly, I really find that this is an area that is often forgotten, and we tend to just use the term idiopathic, which again is not really a diagnosis. You can't call something idiopathic unless you've done all of the standard workup, and the standard workup does include comprehensive genetic assessment. So one thing I will point out here, and we'll talk about this as the discussion goes on, people ask me often who should be referred for genetic testing and who shouldn't. And what I would say for most genetic conditions, even non-cardiac genetic conditions, the likelihood of having a positive genetic test is probably directly proportional to a few things that are very simple to acquire. So if you have a strong family history of a condition where 3, 4, 5 people are affected in the other generations, the likelihood of having a positive genetic test is much higher. Similarly, if the disease manifests at a very young age, then you're also more likely to have a positive genetic test. But like all of medicine, this is probabilistic, and it's very sometimes difficult to really understand. And when you counsel a patient about what the likelihood of a yield is, we often don't know, and so we often quote sort of relatively wide confidence intervals because it can be very unexpected. Some more elderly people can be gene positive, and some very young people with a positive history, family history, can be gene negative. So I usually generally tell patients, if you're younger and you have a family history, we're more likely to find something. If you're older and have no family history, we're less likely to find things. And in general, in most major centers in North America are offering commercial panel genetic testing. So this is next generation sequencing. We only look at genes that are associated with the condition, and we don't look at other aspects of the genome for the most part. There are some major Canadian centers that actually do in-house genetic testing. So the province of Alberta is one, I think Ottawa and Chio as well. So there are some places that do in-house genetic testing like any other lab test, but most major North American centers outsource these tests to commercial companies who, in my mind, generally do a very good job at helping us diagnose patients. And for the most part, again, this is not an absolute, but for the most part, non-ischemic or dilated cardiomyopathies tend to be autosomal dominant in inheritance, and we'll talk about this in a sec. So the next thing is our classification scheme. And one thing that I find very difficult is that when you go through the literature and even just try to do a systematic review or a meta-analysis, there are so many different terms used to describe what is now known as non-ischemic cardiomyopathy. So you'll get familial non-ischemic, familial dilated, idiopathic non-ischemic, inherited, heritable. There's all sorts of different terms that people use, which makes working in this area and the language we use in our clinical letters a bit nebulous. So the ESC has now divided patients into two forms of, quote, dilated or non-ischemic cardiomyopathy. And the important thing to realize is that there's a traditional dilated form of cardiomyopathy where you usually get systolic dysfunction with dilation, and that would be 2 standard deviations larger than norms. But we now recognize as well there's this hypokinetic non-dilated form where your EF kind of drops into the mid-range, but you don't have LV dilation associated with it. And all of the major genes that cause non-ischemic cardiomyopathies can generally present with either phenotype. And so calling it dilated cardiomyopathy is probably a misnomer, although it's a commonly used term. So just be aware of that. So I prefer the term non-ischemic cardiomyopathy, and that's what the European guidelines use. But I also dislike the term non-ischemic because non-ischemic is just ruling out one diagnosis. It's not actually giving a precise classification. So the other principle, I don't think reading through this whole slide is clinically necessary, but one other important principle that I want to bring up that is a challenge in this field is family screening. And what the Europeans have tried to do, which I think is really nice, is they've lowered the threshold for diagnosis in family members of proband cases. So, for example, if your father came in with a severe dilated cardiomyopathy, EF 10%, et cetera, and then you start screening the family and you find a bunch of patients have EF of 50, they have a bit of dilation, but the EF is not less than 50. What the European guidelines are trying to do is come up with a probabilistic classification for relatives where the threshold to make the diagnosis is actually a bit lower, meaning that the family history counts towards making that diagnosis. So I think this is a really nice tool to think about. I don't think it's worth memorizing it, but it gives us a sense of which family members may need to be followed more closely based on their sort of baseline findings of the first assessment. So this was covered very well in the last session. It's the complexity of inheritance, so I'm not going to go into it in detail. What I'll say is most patients with inherited or non-ischemic cardiomyopathies fall into panel B, which is autosomal dominant inheritance with incomplete penetrance and variable expressivity. So in these families, you often see, individuals who carry the gene but don't manifest the disease or manifest the disease much later in life. Or sometimes you see a bit of pleiotropy, which means that, you have a bit of a different phenotype in different family members. So one person may present with heart block first, and another person may present more with left ventricular cardiomyopathy. So we often commonly see that in some of the genotypes. And the vast majority of patients actually have a complex inheritance of their cardiomyopathy. So most of the time, we don't find these rare pathogenic and likely pathogenic variants. In most cases, we actually don't find anything on the commercial genetic test, and that's because there is some form of polygenic risk mixed with environment. And I'll talk a little bit more about that in a few minutes. So this is the same thing said a different way, but if you think about having a baseline risk in the general population of a condition, you have to cross a threshold of disease to manifest the phenotype. So if you have a single pathogenic variant that's very, very high potency, for lack of a better term, you will cross over into the threshold of disease. But these other models really suggest that, you sometimes need multiple genetic hits or, and I'll discuss in a sec, you may have a few genetic hits, but then on top of it, you have some environmental interaction that increases your risk. So to give you a more common situation in which we can all relate to is, you have a family history of some dyslipidemia, which is genetic, but then on top of it, you smoke cigarettes. And some people in the family manifest the disease much earlier simply because of that environmental exposure. And the same thing is probably true in cardiomyopathies as well. So all of these patients have genetic cardiomyopathy, but there's a gradient of genetic risk, which is not always just defined by monogenic variants. So these are the DCM genes that are in the ClinGen summary that Dr. Wilsbacher mentioned. So each gene is assigned a likelihood of being a gene associated with cardiomyopathy. And you can see on the left there's 12 genes that are considered to be definitive and strong based on, published cases and case series and, large multinational studies, but also based on molecular testing, in vitro testing, et cetera. So most of our patients who test gene positive are going to fall into this, but this is an example of the InVite panel. So this was 54 genes a few years ago, but as Dr. Music mentioned, most of the panels for cardiomyopathy are now over 100 genes. So we are doing a very broad genetic test, but 95% of our patients who test gene positive for one of about 8 or 9 different genes overall. Every so often, I'm surprised when I learn about a new mitochondrial gene that's autosomal recessive or something like that I've never heard before, because it's simply a very uncommon cause of cardiomyopathy. Okay. So the next thing I think is an important concept to discuss is when we counsel our patients and, even accept referrals and consider who to send for genetic testing. Like any test in medicine, there's a pre and a post-test probability. So if you limit your clinic to only patients who are less than the age of 30, who have at least 2 family members with cardiomyopathy, et cetera, your genetic yield is going to be 95%. And if your clinic predominantly caters to a more older population, your threshold for referral is very limited. So, anybody with non-ischemic cardiomyopathy, your yield is going to drop significantly. So this is really challenging because every cohort that's published has a bias to it. And that just depends on which patients were actually included in the registry or the study, which directly correlates with the likelihood of having a positive result. So this is a very, very nice paper out of Spain that was published in JACC a couple of years ago. And they attempted to design a risk score to predict who may be more likely to be gene positive and who would be more likely to be gene negative. And they found 5 variables on multivariable testing that had independent prediction of having a positive genetic test. And you can actually look this score up online. It's called dcm-risk.com, I think. Or sorry, this is the Madrid DCM-score.com. And you can click these boxes and it will spit out a number for you. This doesn't mean that there aren't other factors like family history, or family history is on there, but, conduction disease or age or, comorbidities that may play into this. It's just in this cohort, these were the predictors. And you can see if you have 0 points, your likelihood of testing gene positive is very low, probably less than 3%. And if you have all 5, it's basically 100%. So I always make sure in my consult notes and things to mention these risk factors, just because it helps us explain to patients the likelihood of a positive test. And sometimes if patients find out that their risk of testing gene positive is very low, they may choose not to do genetic testing because they don't like the uncertainty brought on by the risk of a variant of uncertain significance. So it's really great to have, I think, a tailored discussion with the patient in front of you about what the likelihood is that we'll find something. So this is from the European guidelines, and I only use the European guidelines because no other guidelines are quite as up-to-date and specific. But it basically says that anybody who has non-ischemic cardiomyopathy should be offered genetic testing, and that we should be using it to cascade family members if you find a pathogenic or likely pathogenic patient. Or you could consider also offering genetic testing in pediatric patients. And the reason why it's only a 2A is that most of these cardiomyopathies don't express in childhood. There are important exceptions, but they only gave it a 2A level of evidence. So moving on a little bit, I want to talk now about a few cases that I think illustrate important points, and then we'll talk about why those points are particularly important. So this is a 40-year-old female who was diagnosed with stage 2 Hodgkin's lymphoma and was started on chemotherapy, including an anthracycline-based chemotherapy. An echo is performed 3 months into her therapy, and her ejection fraction is 40%. Family history is notable for heart failure in her mother and aunt in their 50s, and she is referred to your outpatient office for assessment. So that's case 1. Case 2 is a 60-year-old male who has moderate alcohol intake, intermittent over the course of his life, sometimes more heavy, and occasional cocaine use, who was admitted in cardiogenic shock after many months of dyspnea and anasarca. He is found to have severe biventricular dysfunction with normal coronary arteries. He is diagnosed by the heart failure service with a substance use-related cardiomyopathy and deemed not a candidate for mechanical support or transplant, and he passes away in hospital. 5 years later, his daughter shows up to the office with atrial fibrillation and a mildly dilated left ventricle. And then the third case is a 60-year-old female who undergoes heart transplantation for DCM, and she has a prior history of complete heart block and VT storm. She's discharged from her transplant and put on all the usual medications, and 10 days later, she presents with rhabdo and renal failure. What would the most likely unifying genetic diagnosis be? And so I'll wrap these cases up at the end, but as I go along, I think you'll probably be able to answer these questions on your own. So let's now focus most of the remainder of the talk on rationale for genetic testing. And the question I think many of us often get is, why do we bother with doing genetic testing when you can just screen patients with an echo and an MRI? What does genetic testing give us that clinical testing doesn't? So I want to go through these 7 points carefully and explain each one of them in detail. So the first is that at-risk relatives can be discharged from lifelong follow-up. So the current AHA guidelines emphasize this. It's particularly important to realize that this is only useful if you identify a pathogenic or a likely pathogenic variant in the proband that you feel is the cause of their cardiomyopathy, and then you cascade that specific family variant to the relative and they test negative. Some important caveats are that some likely pathogenic variants may not have as strong evidence as other ones. And in some cases, you find a genetic abnormality that doesn't entirely explain the phenotype or doesn't quite fit. And so sometimes I'm a little bit more cautious about this. But in general, being able to liberate a young person from lifelong echo and MRI and the uncertainties of risk is incredibly important, often for mental health. And I'll also point to our discussion earlier about the discrimination that can exist with doing genetic testing and how we protect patients. But sometimes I tell patients, actually checking yourself for the family variant can actually potentially make you eligible for life insurance when you wouldn't have been otherwise. Because if you test negative and you choose to disclose that to the company, they may recognize that, oh, you're not actually at risk of the DCM that your mom had. So sometimes that can also be very empowering for young patients to have that information. If genetics can't rule out risk in a family member, in other words, the genetic testing is negative or it's a variant of uncertain significance, our guidelines say that every 3 to 5 years, it's reasonable to do an ECG, echo, and Holter. Sometimes I shorten this follow-up or change it depending on the age of onset of somebody with cardiomyopathy, because we don't really understand if, for example, an 80-year-old who develops the condition and is gene negative would have a particularly high chance of passing that risk on to their relatives. But sometimes you have to be nuanced and think about the risks and benefits of screening and also have a shared decision with the family. So the second reason to do genetic testing, and I think this is one of the most important ones, is the ability to predict something that I call LV-threatening events. So this is a wonderful study in JACC from 2018 by James Ware's group, who've done amazing work in cardiomyopathy genetic risk. And what this study shows is that patients who received the diagnosis of alcohol-related DCM were heavily enriched for truncating titin variants. So this gets at the mix of environmental and genetic exposure risk. So I personally counsel all patients who carry truncating titin variants to avoid or limit alcohol as much as possible, because we think it's a direct myotoxin in these individuals. The second thing is that they also found in this study that AF also is much more common in people who use alcohol and have titin variants. So there's other, things that are important to consider in people who carry titin variants. And this relationship about LV-threatening events is also true for peripartum cardiomyopathy and chemotherapy cardiomyopathy. So the first paper is a nice paper from a few years ago in New England Journal, and the bottom one is in Circulation. And these show that patients who have peripartum cardiomyopathies are much more likely to have truncating titin variants, and same with patients who have chemotherapy-induced cardiomyopathy. And as I said, these are mostly truncating titin variants, but other dilated cardiomyopathy variants have been shown too. So I know there's a great deal of controversy in the cardio-oncology literature about who gets GDMT before anthracyclines, for example, and there isn't a great deal of evidence as to whether we should give them. But I have a shared discussion with the patient if they carry a titin variant and need, for example, need chemotherapy. I often tell them it's quite reasonable to start a couple of pillars of GDMT before they start their chemo, because we know that this is a risk factor for expressing their cardiomyopathy. So the third thing it does is it helps to clarify borderline phenotypes. So this is just a quote from the AHA guidelines. And the reason this is particularly important is because in the proband, usually the diagnosis is fairly obvious because they present with heart failure. But in the relatives, it's quite common that the first presentation, when you see them, they have a bit of ST change on their ECG, their voltages are a bit low, their EF is 51, they're heavily athletic, and maybe they have a larger body habitus or something, and you wonder, are these normal adaptive changes or are these pathogenic and signs of early cardiomyopathy? And so genetic testing for the family variant is super helpful in those cases. So I mentioned this a little bit before, but I'll emphasize it more here in point number 4. Genetic testing can inform the earlier initiation of GDMT. This is a nice study from my colleague Christopher Vissing in Denmark that looked at family members who were screened in their clinic, and their first assessment. And what they basically showed is that if there were borderline changes in the ECG and echo on their first screening, they were much more likely to go on to develop familial dilated cardiomyopathy. Whereas patients who had their initial tests were entirely normal had a very low likelihood of going on. So what I do is if I see a family member who's starting to have cardiomyopathic changes and they also have a familial variant, I'm quite aggressive, or at least I have a very informed discussion with the patient about whether or not starting guideline-directed therapy earlier than we normally would might be beneficial. And this is not based on any sound evidence, and I probably will never be, because it's very hard to amass a large enough cohort to actually do a proper study over a long period of time. But this to me, you know, an EF of 50% in someone who has a Titan variant, for example, is very different than someone who has an EF of 50, had a STEMI, recovered from their STEMI, and is now doing well. And the reason I feel that's different is the STEMI is an isolated event that happened once from which you recovered and your scar is basically formed. Whereas genetic cardiomyopathy tends to be a progressive disease over time with the significant risk of worsening. So as people's EF drops below 50, I start to initiate low doses of beta blocker, ACE inhibitor, spironolactone, etc. But again, I tell them that there is the caveat that we're not 100% sure that this works, but I think it's a very logical approach. So the fifth thing is that it can inform pre-implantation genetic testing, and this is just outlining the guidelines, which makes a great deal of sense. There are some of these genotypes that have a very high chance of leading to cardiac transplant very early in life, particularly lamin, which we'll talk about. And so if you know you carry a pathogenic lamin variant, you have the opportunity to eliminate the chance of that happening in your offspring by doing pre-implantation genetic testing and in vitro fertilization. Obviously, across our country and across the world, the availability and funding for IVF is variable, but it's a conversation that I do have with patients when they are in their childbearing age group, just so that they know that that's a possibility. The number 6 reason, which I think is also critically important, is that genotype can inform prognosis. So these are two nice papers. The one on the left shows that if you present with de novo heart failure that's very advanced, can we predict who's going to recover their LV function and who's not? And this paper from the Dutch group shows that if you present with heart failure and you go on GDMT, you have a very good chance of recovering if you have Titan and a very poor chance of recovering if you have lamin. Lamin is defined a lot by heart block and progressive myocardial fibrosis. And so those patients are much more likely to need heart failure or heart transplant and VAD. And so if we're able to get genetic testing quickly enough early in the course of someone's presentation, it may help us, you know, at least be aware to refer people earlier to acute heart failure transplant service to discuss those things, because those things are obviously much easier to discuss in the outpatient setting prophylactically than when there's a crisis and someone's on ECMO. The other thing is that genotype is a stronger predictor than phenotype when it comes to what kind of outcomes patients will have. So this is a really nice paper from the Italian group in JACC in 2022 that showed that genetic testing results prognosticate patients better than any other clinical thing that they could test for. And desmoplakin, lamin, RBM20, desmin, and PKP2 are genes that are associated with very bad arrhythmic outcomes, and lamin has the worst heart failure prognosis. So I tell patients that knowing your genotype does have the ability to tell you what the future may hold. And then the last thing that genetic testing does for us, which I think is an important point as well, is that we often use this term arrhythmogenic cardiomyopathy. And my argument and personal opinion is that all cardiomyopathies are arrhythmic. Some of them are just more arrhythmogenic than others, but there's a considerable genetic overlap between DCM and ACM that's often difficult to tell by just looking at the initial phenotype alone. And I'll explain that a little bit more now. So this is a paper that shows that if you have a truncating Titan variant compared to patients who do not have a truncating Titan variant, those who have a variant are more likely to have ICD shocks than those that do not. Now, Titan is usually considered more of a DCM than an ACM gene, but it's very obvious that Titan is probably actually more arrhythmogenic than gene negative. So to say, you know, some patients with Titan have ACM and some with DCM, I don't think makes a great deal of sense. I prefer to use the term Titan cardiomyopathy, which is much more descriptive. And then the other important thing to realize is that truncating Titan variants independently predict earlier arrhythmias in patients with dilated cardiomyopathy in this study. So again, even though we think Titan is a DCM gene more than an ACM gene, it is certainly more arrhythmogenic than the run-of-the-mill non-ischemic cardiomyopathy that's gene negative. Transitioning on from there in a similar light, I want to talk about how genotype can actually do a very good job of predicting what phenotype we see. So there are some genes that are much more associated with arrhythmogenic left ventricular cardiomyopathy or left ventricular DCM, and some that are much more associated with ARVC, or arrhythmogenic right ventricular cardiomyopathy. So knowing someone's genotype helps you decide and know what they and their family members may be at risk for. And the other thing that's very helpful is that many of these genotypes are actually subtly syndromic, meaning that there are characteristic signs beyond cardiomyopathy that help you know what you're dealing with. And I'll talk about a few of the more obvious ones, but some of them are cardiac manifestations like heart block, um, some hypertrophy, and some of them are extracardiac, like skin and hair and nail changes, muscle changes, things like that. And it's critical to make these diagnoses because your threshold for ICD changes based on your genotype. So as you know, the Heart Rhythm Society has specific ACM guidelines. They used to be ARVC, but Heart Rhythm switched to talking about ACM. And what they say for the ACM genes, which are the ones I mentioned were the most arrhythmogenic, we actually follow patients more closely for a rhythmic outcome. So you'll look here, the other guidelines said for DCM, we screen with those tests every 3 to 5 years, but these guidelines say if you have a very arrhythmogenic cardiomyopathy, family screening should be every 1 to 3 years, and it usually starts earlier in life. So the gene really informs how closely you screen relatives. So now the last thing I'm going to do is move on to a few classic genotypes that everybody should be aware of and how we risk stratify them in the modern era. So I'll only have one slide on ARVC because it's not traditionally thought of as a non-ischemic cardiomyopathy, although it overlaps heavily sometimes with the arrhythmogenic cardiomyopathies, and you can have left-sided involvement. So the one form of ARVC to really know well is plakophilin-related PKP2. And this is a gene that is responsible for making one of the desmosomal proteins, which are the sort of glue that hold cardiac cells together. These patients tend to have classic ARVC, so they don't usually get left ventricular involvement until very end-stage disease. And they have classic fibrofatty replacement of the free wall of the RV. It typically presents in younger individuals, and especially in those who do endurance sports. So the chronic volume loading of the right ventricle leads to earlier expression of disease. And so this is the classic disease that affects young marathon runners who have like sudden death during exercise. And the reason why they first present with sudden death is that the right ventricle is highly malleable and adaptable. So before they start developing right heart failure, they actually get arrhythmias first, as opposed to somebody who has more left ventricular involvement. Those patients, once their EF drops to 35-30, they start to get heart failure symptoms before they get a lot of arrhythmias. That's not always the case, but in ARVC, sudden death is such a problem because patients don't have symptoms from RV failure early on. And the nice thing that we now have is, thanks to Giulia Cadrantorini in Montreal, is an ARVC risk calculator where you can type in a number of variables online and it will spit out a probability of a life-threatening ventricular arrhythmia or sudden death within 5 years, and it can help you decide who gets an ICD. The next cardiomyopathy I want to talk about is desmoplakin. Desmoplakin is a fascinating cardiomyopathy because the DSP gene is expressed in skin and cardiac cells, and it's similar to PKP2 that it forms the desmosome, which is the cement that binds the bricks together. And so patients tend to get a myocardium that starts to essentially fall apart, but they also get skin and hair and nail changes quite early in life sometimes. Not every patient gets these, but some patients do. And so I always ask patients with cardiomyopathy about a history of very woolly or curly hair. Many people have such curly hair that they actually straighten it, so you actually have to ask about it. And then I also ask to see their hands and feet because they get very calloused hands and feet oftentimes. And you can actually, in a large desmoplakin family, often pick out who's a gene carrier or not simply by looking at these things. And many patients will have actually seen a dermatologist before they see a cardiologist. So these patients present in young and middle adulthood. There's no clear association with endurance sports. And they also have a very interesting phenotype where they get these hot flares, where they get almost like a myocarditis-like presentation, and they come into hospital with troponin elevations. They often get coronary angiograms and MRIs. And we think those myocarditis flares are just periods of time where the myocardium becomes inflammatory, and we don't quite understand, but it is closely related to desmoplakin. And the latest studies show maybe 1 in 4 patients have this manifestation. They are quite arrhythmogenic, so we have a lower threshold for ICD, and we actually now have a wonderful DSP risk score based out of Johns Hopkins, where you can again type in patients' risk factors and calculate their 5-year risk of ventricular arrhythmia. And then the last cardiomyopathy I want to talk about is lamin. Lamin encodes the lamin AC gene, and it's an nuclear envelope protein that's expressed in both cardiac and skeletal muscle. It's actually ubiquitously expressed in many parts of the body, and it's associated with many other non-cardiac diseases as well. These patients present very early in life often. They have a lot of arrhythmias. They have very difficult control atrial and ventricular arrhythmias. They can still sometimes get complete atrial standstill and progressive heart block. And it forms a group of conditions called the laminopathies. These patients usually require heart transplant. It's a highly penetrant disease, and they have an earlier indication for ICD. We also have a nice lamin risk calculator as well online that you can use to calculate their risk of needing an ICD. And then the last thing I want to talk about is the fact that MRI is extremely valuable in these conditions. And this is the guideline paper from the Europeans, but what it shows is that each genetic cardiomyopathy actually has a characteristic MRI pattern. And you can actually predict what the genetic testing is going to show based on the MRI. And our cardiac radiologists at St. Paul's are so well aware of this and up to date on the literature. They'll actually often tell us that they think they see desmoplakin-like disease, and then the gene test is positive. So I use MRI in all of these patients, and I follow MRI every 3 to 5 years if they don't have a device in, and I find it to be very helpful. And again, it argues in favor of classifying based on genotype and not phenotype, because each of these have a very clear phenotypic signature. And our guidelines recommend using MRI. So I'll just wrap up these cases. So case 1 was an individual who had chemotherapy-induced cardiomyopathy and a family history of heart failure. This is truncating titin cardiomyopathy that's unmasked by chemotherapy. The second case is a titin case that's unmasked by a substance use disorder. And then the third case is this individual, unfortunately, who has lamin cardiomyopathy. And the reason she developed rhabdomyolysis is because she was started on statin therapy to prevent coronary artery vasculopathy after a transplant. And lamin is a cardiac and skeletal muscle disease. And although the skeletal muscle is often fairly mild and all they have is a bit of a CK elevation, if you give them drugs like statin, they can have severe muscle breakdown. So her diagnosis was actually made only after she underwent heart transplant. So this is just a concluding slide. I'm not going to reiterate all of these things, but I'm always happy to share my slides, and it's a sort of a nice summary of, I think, everything you need to know about the basics of genetic cardiomyopathy, how to diagnose, how to manage, how frequently to screen patients, and when to consider starting guideline-directed therapy. So I'll stop there, and I look forward to your questions and the discussion to follow.
Wow, Thomas, thank you. That was incredible. I have a number of questions, but I see we already have some in the chat here, which we'll go through. But I did just want to make one reflection first, was that how unfair or unjust it is that an individual would be labeled with a term substance cardiomyopathy or substance-related cardiomyopathy when they had a TTN-related cardiomyopathy all along. So thanks for highlighting that. We'll start with a great question by Dr. Swigum. Did you, Liz, if you're on, do you want to just ask the question? It might be nicer rather than us reading out.
Thank you. Thank you for calling me out.
I'm sorry.
This is an amazing presentation. Thank you, Thomas. The question that I had was just to relate to the resource scarcity throughout Canada. And knowing when we have an undifferentiated non-ischemic cardiomyopathy, again, to your point, we've ruled out coronary artery disease, and there's scar on cardiac MRI. You know, usually there's a little bit of a cascade in terms of the screening test. And my question to you is, from your practice and from just the right thing to do perspective, should we be doing medical genetic testing concurrently with some of the other imaging? For example, if there's scar, we are looking for cardiac sarcoid and medical genetic testing at the same time, because that cascade screening can sometimes take, and I had said 6 to 18 months, but for some places it may be longer or never.
Yeah. Yeah, I think Dr. Swigum, that is a very good point, and I totally agree with you. So often genetic testing is the last thing that patients go through, especially in Canada. And they have all these crazy things, including like a biopsy that's not clearly indicated, and then finally 5 years later they end up on the genetic doorstep. And I found, where I've trained previously, particularly in the US, genetic testing was actually one of the easier things to access, and it's the opposite. And I think, the 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 pretest probability of a genetic disorder is way higher than isolated cardiac sarcoid. So we have it backwards in Canada. And I would argue that PET is actually a more precious resource than genetic testing. It's just, I think part of it's the way we think as cardiologists, and part of it is the system that we work in.
And could you also expand on that, the cost saving that comes with genetic testing in terms of dismissing variant negative family members?
Yeah, I mean, for sure. I mean, when you think about screening a 20-year-old every 3 to 5 years with an echo and MRI or Holter for the rest of their life, and you think about the cost of parking, them taking time off work, them, thinking about the stress of starting a family, their risk, alleviating someone of that 50% of the time is a hugely powerful tool. I would say we don't have anything else like that in cardiology, but we just don't do it as often as we should.
Okay, thank you. Sanela, do you want to?
Yeah, there's another interesting question in the chat on a little bit hip-hopping on the alcohol and the chemo-mediated cardiomyopathies. If there's a rate of underlying genetic mutation that's been looked at or established and what has been previously labeled tachycardia-mediated cardiomyopathy, speaking perhaps of the overlap of AFib and arrhythmias with cardiomyopathies in genetic conditions.
Sorry, I'm not. Was that a comment or a question? I missed.
It's a question. If there is an established connection or an established rate of genetic mutations that are identified in what's previously been labeled as a diagnosis of tachycardia-mediated cardiomyopathy.
Yeah, I'm not sure of any literature I know specifically on that question. There may well be, but there's a lot of studies now looking at, so there's a study looking at what's the rate in chemotherapy, what's the rate in alcohol. There's one on, what's the rate in isolated cardiac sarcoid. And I think it's only a matter of time before we figure out what the rate of positive genetic testing is in tachycardia-mediated cardiomyopathy. But I will say in all of that, those percent yields entirely depend on a referral bias. So all of these patients in whom the cardiomyopathy is not as well as explained as we think it should be, and that's clinical judgment, we should offer genetic testing. So, if you've had, huge high doses of anthracyclines over a long period of time, that's very different than someone who has modern chemotherapy that's dosed very low. So there's a clinical judgment that comes into all of it.
I'm going to ask sort of a counterpoint or question, because I imagine everyone's minds are racing now. Would you, Thomas, say that someone who is about to undergo potentially cardiotoxic chemotherapy should be screened with genetic testing to determine whether they have a Titin variant and what their risk is? And in answering that, could you just tell us like what the general population, like the population frequency of Titin variants are? Because I think that would be surprising to us.
Yeah, great question, Omid. So about 1 in 200 to 1 in 300 people in the general population have a truncating Titin variant. They are associated in the large, UK biobank studies. It actually is associated with a statistically significantly lower EF. It's just that when it's discovered incidentally, like 95% of people still have a normal EF. It's just lower than population average. So instead of it being 57, it's 53. I think, that genome first approach to medicine that you talk about isn't just applicable to cardiomyopathy. So, in some places, big centers in the US, the Geisinger comes to mind. They actually do genomics on every patient. They do a whole genome on every single patient who comes through, and they predict their pharmacogenetics. They predict their risk of long QT. They predict, all these things that may be modified if you knew someone had a long QT variant and you don't give them moxifloxacin for their pneumonia. So I think there's an argument more and more towards being genome-based first and testing these things, but you have to keep in mind the downside, which is the risk of unnecessary cascade testing, because all of us, on average, have 8 highly, highly rare genetic variants that could be classified as damaging if we did our whole genome. So if you're very agnostic to a phenotype, you are more likely to drive downstream testing and anxiety than not. So there's a positive and negative side to all of this.
That's great. Dr. Wilsbacher gave an example of where, I looked at Duchenne as sort of the prototype of a genetic cardiomyopathy with basically 100% penetrance and expressivity. And in that population, she described how treating with medicines, heart failure therapies, particularly ACE inhibitors, is life-saving if initiated before the onset of a cardiomyopathy in a variant positive individual. So how would you suggest that we follow patients who are picked up through predictive testing? So they have a family member who's got a cardiomyopathy, and then they learn that they have the same variant, they have the same gene. At what point do you, and you touched on this a little bit, but at what point do you start medical therapy for them?
So I have a shared discussion. I know that I'm not saying that because I don't have an opinion. I have a shared discussion about where they are in life, what their priorities are, how old they are, what their other comorbidities are. So, actually, like in you,
we're just cutting out a little bit there, Thomas.
Pril and spironolactone, bisoprolol. And if you're younger and you're of childbearing potential or thinking of having a family and you're female, maybe we won't give you ACE inhibitor right away in life, for example. So I think I start to have the conversation when the EF is less than 50. If the family history is very bad or the genotype is very bad, then it's quad therapy once the EF is less than 50. But in some patients, they only want to take an ACE inhibitor because they hate beta blockers, and I say that's fine. And some patients choose not to take it, and I just follow them more closely.
That's really helpful. Well, thank you. I think we're at time now, just because we have to join the closing remarks by Dr. Burney and Patterson. I really want to thank not only our speakers who were amazing, but everyone who took the time this afternoon to join us to learn about these important topics. We hope to see you next year and are very grateful for your time. So thank you.